Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Welcome to visiting lecture, Advanced Research on Chemical Biology Interface for Sustainable Development 2023. It is my pleasure to welcome you to this event. Allow me to introduce myself. My name is Rahmadillah Khairiyah, and I will be your master ceremony for today. On behalf of the Diponegoro University Chemistry Department, we extend our appreciation for your presence today. In this event, we will have several sections. First is opening speech, the second is speech from our speaker, and the last session is discussion and Q&A. Now, we are going to have a really incredible session. So I hope you guys can put full attention for all our speakers today. Hello, Dr. Cahyo Budiman, Dr. Ruzaidi, Dr. Khairul, and Dr. Mardani. Welcome. Ladies and gentlemen, before we commence our event, let us now stand together to sing the Indonesian national anthem, Indonesia Raya. To all participants can sit down. Thank you. Let's begin this event by opening a speech from our chemistry department chairman. Please welcome Mr. Adi Darmawan, PhD. Time is yours.
Assalamualaikum warahmatullahi wabarakatuh. First of all, I would like to welcome Dr. Ruzaidi, Dr. Khairul, Dr. Mardani, Dr. Cahyo. Welcome to Undip, to Faculty of Science and Mathematics, and to Department of Chemistry. Actually, this week is very hectic. We have 35 FIFA in this week here, so maybe some of the lecturer can't come to this event because some of doing examination in FIFA and some is doing lecturer. So first I would like to very welcome to you the relationship and collaboration between Undip and University Malaysia Sabah is very close. Bu Meni already come to University Malaysia Sabah. Dr. Ruzaide also have given lecture last year when you were in airport I remember that you are in the airport at the time. Yeah. Because you are doing online webinar. Yeah. In February also Professor Zazmal and Professor Zarina from, I forgot, is from also in the same department in this same institute. Yeah. They are gifts, so also visiting lecturer to us. So this week also 3 of our students will are going to OMS to do how to say no internship, just like for change, student exchange for 1 semester. So I hope that maybe one time some of the UMS student can come to here. Yeah. I hope that the relationship between Undip and UMS can extend to the another to future to more intensive collaboration. I hope that you can enjoy here in Semarang. Maybe if you have any time to just go around to just to see, saying to Semarang. It's very hot. Semarang is very some people say that Semarang is between the sun and the earth. So so this is the sun, this is Semarang, and this is the earth because it's very hot. But yeah, but this is Semarang. This is nice city. Thank you. Maybe this from me. I hope we can get a benefit from the all the lecture today. Yeah. Saya akhiri. Assalamualaikum warahmatullahi wabarakatuh.
Thank you. The next session is a greeting that will be delivered by our Dean, Faculty Science and Mathematics, to Prof. Widowati, MST. Time is yours.
Thank you very much, Master of Ceremony. Assalamualaikum warahmatullahi wabarakatuh. And good mornings, everybody. Excellencies, speakers, Dr. Ruzaidi Alimoh Mokhtar, and then Dr. Mardani Abd Halim, and then Dr. Khairul Azwar Kamaruzaman, and also Dr. Cahyo Budiman. I already met maybe with Dr. Cahyo. Okay. Honorable Prof. Adi Darmawan as a head of chemistry department, Prof. Maini Suseri, the initiator of this visiting lecturer, manager and supervisor, and then lecturer at Department of Chemistry and all participants. First of all, let us give thanks for the presence of Allah Subhanahu wa Ta'ala, the most gracious and merciful God, because with his permission today, we can gather here to join together for the visiting lecturer events. Ladies and gentlemen, it is my great pleasure to cordially welcome you to the visiting lecturer events. And then the topics of this event is advanced research on chemical biology interface for sustainable development. This event organized by the Chemistry Department, Faculty of Science and Mathematics, Diponegoro University, and an integral part of our fruitful collaboration with the Biotechnology Research Institute of University Malaysia Sabah. The aim of this visiting lecturer is in line with the goal of the Faculty of Science and Mathematics to facilitate brainstorming and state-of-the-art information in the fields of sciences and mathematics. As we know that the Faculty of Science and Mathematics Diponegoro University always supports research and education development in mathematics and its application. Several visiting lecturer programs, young professor, student exchange, and international conference have been implemented to promote Diponegoro University as a world-class university. We also support our students to gain an international atmosphere by conducting research and short-term study. In the future, maybe we have international undergraduate program of chemistry bachelor can study in the Sabah University, Malaysia. And maybe student exchange of master degree in the chemistry students. We always welcome international cooperation and collaboration to enhance mutual advantages in research and education. I think there are many publications have published, especially with author or co-author for our professor from chemistry department, especially Professor Manny. Thanks to Professor Manny. Give applause for him. Ladies and gentlemen, our collaboration with the Biology Research Institute is a testament to our commitment to advancing science knowledge and promoting cross-disciplinary partnership. It is commitment to harness the collective power of our intellectual capital to address complex issues that extend beyond the boundaries of any single field of study. I also would like to express my appreciation to the head of departments chemistry and also to Prof. Manny Suseri, who initiate these events, and all of organize the team and everyone involved in making this event reality. Your hard work and dedication have made today's lecture possible, and we are all beneficiary of your effort. Finally, by the grace of God Almighty, consent of all participants, on behalf of Faculty of Science and Mathematics, Diponegoro University, I am as a dean hereby announce that visiting lecturer event with topics advanced research on chemistry biology interface for sustainable development is open. I encourage all participants to participate actively in the interesting discussion. Hopefully, what we are doing today is useful for our progress in the future. Thank you very much for your kind attention. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you. The next session, ladies and gentlemen, please join me in welcoming the representative from Faculty Science Mathematics Universitas Diponegoro and Biotechnological Research Institute Universitas Malaysia Sabah to start for the signing, memorable of agreement and implementation arrangement ceremony. This momentum occasion symbolizes the beginning of journey that hold great promise for the future. To Dr. Rozaida and Prof. Widowati, please welcome. Give applaud and congratulate to Faculty Science and Mathematics and Biotechnological Research Institute University Malaysia Sabah. Okay, we continue to presenting appreciation souvenir from Faculty Science and Mathematics, Diponegoro University to University Malaysia of Sabah. Okay, next we continue to documentation session. To all participants, come forward to take some photos. Please come forward. Come on, guys. Okay. Ladies and gentlemen, let's continue our session for Studium General. Okay, I would like to introduce our moderator today, Dr. Agustina. She's one of the lecturers that expertise in biochemistry that have recent interest in natural product from biotransformation and also working in field of endophytes. Without further ado, let's start our session by welcoming our moderator. Please welcome Dr. Agustina. Time is yours.
Okay, good morning. Assalamualaikum warahmatullahi wabarakatuh. And all of the sekalian, ladies and gentlemen. Thank you very much for coming to this event. So I'm professor. Sitting lecture. We are going to have 3 lectures. Start by Dr. Last 30 minutes, we are going to have ID. Curriculum vitae of Dr. Ruzaidi. He graduated from Applied Biological Science, Tokyo University Agriculture in as a bachelor, and then master from Nutritional Biochemistry from University Putra Malaysia, and then his PhD in Physiology from Monash University, Victoria, Australia at 2016. The professional appointment, especially as a Deputy Director in Biotechnology Research Institute, University Malaysia Sabah, and his publication until now is 25 articles under Scopus Index with each index of 12. So without further ado, please Dr. Zaidi, he will giving lecture about exploring the therapeutic potential Sabah mangrove plants as anti-tuberculosis and anti-dengue agents.
Thank you, Dr. Christina. So how I'm going to share the slides? Just share here.
Just share using the
Assalamualaikum and very good morning, everybody. My name is Ruzaidi. So first of all, I would like to thank you to Faculty of Science and Mathematics, Undip, for inviting me to these inviting lectures. Actually, this is I think it's third time for me with Undip. First time is back to 2017. When I come with Pak Chayo, during that time, it's still old buildings. This new building still not complete, I think. Okay, and then the second time is last year through webinar, and then this third time. Okay. So before I start my lecture, is allow me to introduce a little bit about our institute for those that is not very familiar with Biotech Research Institute. So just give me about 10 minutes to introduce our institute. Okay. So this is our institute. We established in January 2022. Our institute is one of the 4 centers of excellence in UMS. In UMS, we have Institute of Biological Tropica, Marine Borneo, and also the Boris. What is Boris? Humans study, something like that. It's not science. So we are one of the 4 centers of excellence in UMS. Okay, this is not really new buildings. I think Ibu Maini already went to our institute. So this building is completed 2011. And this is our current director is Prof. Dr. Lee Ping Ching. I'm no longer the deputy director. Now the deputy director is Dr. Kenneth and Dr. Mylene, and assisted by Dr. Nur Atira as a postgraduate coordinator. So I just go very quickly. This is our lecturers in IPB. In total, we have here Dr. Chayo, Dr. Karo Asfa, and Dr. Mardani. Okay, and then in total we have 22 academic staff, and non-academic staff we have 21. This is quite balanced between academic and non-academic staff. Okay, here we also have our officer here, Mr. Haslan, join us to help us in all the admin things lah. Okay, so our focus basically we are not a faculty. That's why we more focus to postgraduate teaching. So this is automatic. Under institute, we only have the postgraduate program. We don't have an undergraduate program. We have actually, this not in the slide. We have 3 postgraduate program, which is the Master Science in Biotechnology, and also we have which is S2. We have also PhD in Applied Biology, which is S3. Currently, we have around 80 students. Master is about 40 to 50, and we have about 30 PhD students. Just go through quickly. This is our main focus, research focus in Biotech Research Institute, which is agro biotech, healthcare biotech, and also the industrial biotechs. Under our institute, we have 7 or 6 research. We call it research group, head by. So here we have head by certain lecturers. We have microbiology and tissue culture, natural products and drug discovery, molecular genetics, molecular microbiology, bioprocessing, and it's a biosensor. Okay, this is the facilities. I think Bu Maini is quite familiar with our facilities. We have we are not that big. We only have about 10 main labs. Our lab is more to the research lab. It's a lot of equipment and so on. And also we have we're proud we have the biosafety level 3 labs. So if you're working with a pathogen level 3, for example, me working with tuberculosis, so we need to do our research in this lab. and we have several support facilities. So this is the example of the lab. In the lab, this is in the genomic lab. We have the sequencer, PCR, and so on. And this one is the most expensive lab that we have. We have the LST QTOF, GCMS, ICPMS, and also NMR is the most expensive equipment we have. Okay, and so on. This is all the other facilities like seminar room, and so keep going. Okay, this a little bit about our achievement. Yeah, last year we published about 62 papers in index journals, and then this is the amount of grants is about 5 to 6 million every year, and this is our collaboration. So if you can see here, we have the Undip here. Okay, I think that's it. I think this is all the MOU research and so on. Okay, if you're interested, especially for the student, if you're interested to maybe to further or to attach with biotech research, you can contact us at any time. Even though if you want to go further your study in S3, PhD, yeah, we are more welcome for the students to come. Okay, I will proceed with my presentation. Okay, so my presentation is entitled "Exploring the Therapeutic Potential of Sabah mangrove plants as anti-tuberculosis and anti-dengue agents." So this project is a collaboration with the Indonesian university, which is the Swiss German university. So we've done this one, I think, since 2018, yeah, with Swiss German university. Okay, as the introductions, we all know mangrove. I think you all know mangrove because, as you know, that Indonesia is the largest mangrove area in the world. Okay, so but in Malaysia also we have a lot of mangrove. So I'm not sure in Indonesia what you call it, but I think it's the same, bakau. Yeah, these trees is along in Southeast Asia is quite common. So why we choose this plant actually is through our collaborator in SGU. They done the research on antibacterial activities. They found that the antibacterial activities in mangroves are very high. Some of the result is higher than the commercial antibiotics. So that's why we choose mangrove as our sample, especially in Sabah. There is no studies done on mangrove. Okay, just a little bit about the global distribution. As I said, Southeast Asia is the largest mangrove area in the world, and Indonesia is the first. I think Indonesia cover about 30% of the mangrove area around the world, and Malaysia is second in the Southeast Asia. In the world, I think 6 or 7, I'm not sure. But it's, what I want to stress here is in Malaysia, Sabah, where is our university located at, is the 60% of the mangrove area in Malaysia is in the Sabah, more than half in Sabah. That's why there is no study about the mangrove. That's why we are the pioneer that explore the properties of mangrove, especially towards the human disease. Okay, this is some of the medicinal properties of mangroves that been studied previously. So most of the studies done in Northeast in Asia, most of it done in Brazil. So are reported to have the bioactive compound and also have the antioxidant activity, antibacterial, antiviral, and so on. So that's why, but for this study, we just focus more to antibacterial and antiviral. So we choose one is antiviral. The common virus in Southeast Asia is a dengue. So that's why we choose a dengue. And then the other one is for bacteria is we choose TB because I think Indonesia is the highest TB incident. I think not the highest, among the highest. I think second in the world. Malaysia also is not that high, but particularly in Sabah, we are the highest in Malaysia. So that's why we choose TB and also the dengue. So a little bit background about TB. It's a second leading infectious killer. So worldwide, we have about around 10 million cases every year. And then the death is about 1.6 million. And as I said, Southeast Asia is the main contributor to TB cases in the world, which is 43% of TB cases is come from Southeast Asia. And then I think Indonesia is the second large contributor to the TB cases. And I said just now, in Malaysia, the incidence rate is quite low. But in Sabah itself is 128 cases per 100,000 population. But Indonesia, of course, is 3 times higher than Sabah. Okay, the problem of the treatment, because this disease is mainly in Southeast Asia. I think the European don't interested to study this disease because this is not their problem. This is our problem. So that's why we need to find the alternative or the drugs that we can treat this TB. So there's a lot of current treatments. There's a lot of problems. I don't want to touch on that. For example, multi-drug resistant and also and so on. Okay, about dengue, as I said, dengue also is quite problems in our region. So especially in Malaysia, I think the dengue cases is higher than Indonesia. But I believe that Indonesia also dengue cases is quite common, occurs here. So the problem for dengue is currently is no vaccine and treatment for dengue. So that's why we hope that with this study we can find certain compound that can treat or that can inhibit the dengue virus. Okay, for this presentation, I divide it into 3 different studies. So the first study is more to we analyze the total phenolic content and antioxidant of the selected mangrove plants, especially from Sabah. And then the second study is we focus more to anti-TB activity. And then the third study is more to anti-dengue activity. Okay. Okay, this is the sampling site. For those who don't know where is Sabah, we are located at north of Kalimantan. Okay, here. So our sampling site is near our university, which is in Tuaran. It's about 30 to 40 kilometers from University Malaysia Sabah. It's quite close. And this is where our samples is where is the sample collected from the Sulaman wetland. This is just a picture of the Sulaman wetland. It's a very beautiful place. So most of the sample is collected around the office of this Sulaman wetland. This is just a picture. My students collecting the samples. So some sample we need to go by boat inside the forest to collect because it's not available near to the office. Okay, this is the samples that we choose for the study. So the justification why we choose only these 6 species, because this is because of we select it based on the report from our collaborator in SGU, saying that these species have very high antibacterial activities, and also the availability of these species in the Sulaman wetland. So in total, we have 6 species and 3 different parts. So if you can see, some species we don't collect the stem, some species we don't collect the root, because this is based on the SGU's recommendation. Only this part is have the high antibacterial activities. Okay, for the solvent, we use different polarity of solvent. We cover from polar, semi-polar to polar solvents. So in total, we have 52 samples. Okay, this is the process. I think you all may be familiar with the process. Collect, wash it, cut into small pieces, and then dry it. And before we ground it to be a powder, so much more easier to extract this plant. So as I said, we use 4 different solvent here. So we use the maceration techniques. After macerate, we just filter it and then remove the solvent, and then dry it, and then keep the extract in the fridge for further use. Okay, this is the method. It's quite common method. I think maybe all of you are quite familiar with the total phenolic content. It's quite a general method using the following Kyokaltu reagent. And then for the results, we can see that the ethanolic extract normally have a higher total phenolic content compared to the other solvent in general. So we have here aqueous ethanol. The ethanolic extract normally we have about 300 to 400 milligram per gram phenolic. And compared to ethyl acetate, also it's not that high. It's about 200. And of course, hexane is much more lower because it's a non-polar solvent. Okay, that's the about the total phenolic content. So for antioxidant, we use the DPPH scavenging assay. This is also a common method for antioxidant. And then as a result, again, the same trend, the ethanolic extract show higher antioxidant activity with the lower IC50 value compared to the other solvents. Okay, so I think there's a lot of papers already reported that the possible relation between the total phenolic content and also the antioxidant. Because normally most of the antioxidant activity is contributed by the phenolic compounds inside of the plant. So I think our research also support that statement. So most of our sample that have a high total phenolic have also high antioxidant activity. So and then between the parts, higher TPC and antioxidant is showed in the leaf and also stem, basically. Okay, so that's about the total. I don't want to go further about this because one more focus about the TB and anti-dengue. So we go to the second study. It's about TB. I'm not sure. Okay, for the anti-TB activity, we use the Resazurin microtiter assay. This one for sake to screen the 52 samples to find which sample have the most or the highest anti-TB activities. So this this screening also is quite easy actually. So we just monitor the change of color from the blue, which is the Resazurin color, to the pink color. So indicate that there is a TB bacteria in the culture. Okay, this is the method. Again, I'm not going to detail about the method. So all of this is done in the BSL3 lab. We cannot, I'm not sure in Indonesia, but in Malaysia, TB is categorized as the level 3 pathogen. We need to do all this experiment in the biosafety level 3 lab. So as you can see, up there is my student doing the lab. We need to wear all the suit and everything. It's not convenient to do research with all helmet and so on. Okay, basically we incubate the bacteria about 3 to 4 weeks before we put it in the well for experiment. This is a TB look like in the tube. So maybe you cannot see here. If you can see the white crumb here, that's the TB. TB bacteria. Okay, after we culture it and then we transfer it into the 96 well for screening. So basically, we will incubate for 5 days in the 96 well before we put our sample, and also the positive control here. We use the rifampicin to see the color change from blue to pink color. So to determine the IMIC value of the samples. Okay, this is the much larger picture. Maybe it's normally in one 96 well we can only test about 3 samples. That the maximum that the maximum we can do. So here is a okay. It is 3 samples here, and then we need to have the growth control, which is the medium and also TB. So here we can see definitely we need to see the change of color from blue to pink to make sure that the TB bacteria exists in the well. So this is the drug control. As I said, we use the rifampicin. So of course the drug will inhibit the growth of the TB, and the blue color is remain. And also this one just sterility control to make sure that all the well is sterile. So okay, from this we do the serial dilution. So after that we determine the MIC. So if you can see here, because we need to incubate this well about 1 week. Yeah, so that's why we put the water at the outer parameter here to make sure there's no hydration. Yeah? So make sure that there's enough water surrounding, because we put in the incubator, it's quite dry. So that's why we need to put water here. Otherwise, the media will dry out and then the bacteria will die. So this is how we do the screening. And then again, okay, so just go to the results. So as you can see here, as a result, if in the aqueous extract, the MIC value is between 2.5 to 5. Ethanol is 0.625 to 125. It's quite high. Basically, I will show you later when I compare with other studies. Okay, this is how it really look like. So we determine the MIC value based on the the lowest, not the low, the higher concentration of apa, yeah, macam mana nak cakap tu? Yeah, the lowest concentration of the plant extract that can kill the TB bacteria. So that's a MIC value. For here is ethanol is there, but surprisingly, actually, it's not ethanol the highest. Even though the total phenolic and antioxidant content, most of the sample in ethanol is high, but MIC is okay. It's quite high, but surprisingly, actually, our result shows that the ethyl acetate extract have the highest anti-TB activity compared to other solvent. So here we have the syrup tagal. This one I think leaf, which the MIC value is 0.04. Is very very low and is very very promising so that we can further study using this sample. So I will show you after this. We select this syrup tagal leaves for the in vivo study, which is we use animal, we infect the animal with TB, and also study the infection, the TB gene, also the immune gene of these animals. Okay, just to summarize the in vitro study, the highest is in the syrup tagal leaf. It's very strong. As I said, if we want to compare with the previous study, for example, here the study by Jan Manchi in 2017, it is ethyl acetate fraction from the Angiogyrus corniculatum, which is also the mangrove species. The MIC is 6.25. Compared to us is 0.04. It's very, very low compared to their study. Most of our extract, the MIC is the lowest, the highest is 5, even lower than this study. So we can say that all the extract actually have the anti-TB activity, but the highest is the syrup tagal leaf. So that's why for the further study, we choose this syrup tagal leaf for the in vivo study. So as I said, this one is a very comprehensive study, but I'm not going to go through everything. So as I said, here we infect the mice with the TB through the intranasal. And then here we have two types of treatment study. First is treatment study. Why we call treatment? Because we infect the mice with TB, and then we give the extract as a treatment for 14 days. And the others, we have the protective study, which is for this study, we give the extract first for 2 weeks, and then we infect the mice with the TB, and we see whether the extract have the protective effect or not towards the TB infection. After that, we will take the lung, harvest the lung, and then do the RNA extraction and do the PCR to see whether there is infection or not. For my student, they go for more further study, which is use the immune gene. Yeah, they have I think more than 10 genes that represent the innate and adaptive immune gene. But for this presentation, I just focus more to infection gene. Yeah? Okay, so this is the timeline of study. As I said, for the treatment, we infect first, and then go treatment, and then but for protective, we give extraction first before infect it with TB. This is the group. So the concentration that we use is only 200 milligram per kg. So yeah, we have justification why we use this because we have study, means we do the toxicity study and so on. That's why we at the end we decide to choose the 200 milligram per kg body weight, the concentration. Okay, just to show that here, okay, after TB infection, actually, you can see the sign of a TB infection in the mice. So here, one of it, we monitor the body weight of the mice. So if you can see here, the red box here, which is the untreated, which we don't treat these mice with extract or the drugs. You can see the significant reduce of body weight of these mice if you don't treat it with drugs or sample. Okay, this is quite obvious sign of TB infection. And also this one is for the protective, is same. There's a reduction of body weight in the untreated group. Okay, significant weight losses about 9 to 13% from day one. So it shows that we are successful infected the mice with the TB. Of course, after this, we do the PCR to make sure that the infection is successful. Okay, but with the treatment of mangrove extract, which is Citroptagal leaf, so we can see that the body weight is back to normal. So that means the extract have ability to treat the infection or to protect the infection. So we can see the difference between treatment groups with the control group is not significant. Means the body weight is back to normal. Okay, this one is both for the treatment and also for the protective study. Okay, this one we go further and we extract the RNA from the lung of these mice, and then we just to make sure that is it really the infection is really happen in these mice. So of course, after we test it, we use the TB gene, which is IS610 and also MPB64. This is the most common gene to detect the infection of TB in mice. So as you can see here, compared to the healthy mice, there's a upregulation of this gene in the TB infected mice. So it's very high. Okay, I don't want to go this one. This is the gene, immune gene that my students done. Also, if you can see, the immune gene also is upregulated. This is quite significantly. Okay, we go for the. Yeah, this is just summary. The upregulation of Mycobacterium tuberculosis and immunogenes in untreated shows that this infection is successful. Okay, this is for when we compare the untreated mice with the treated mice. So here we can see that the mangrove extract is able to down-regulate the infection gene in TB infected mice. You can see here, so both genes is down-regulated after we treat it with the mangrove extract, which is a Citroptagal leaf. And this for immune gene also, basically all the gene is down-regulate in the treatment. Okay, and then again, same what you call it, same pattern we can see in the protective study, which is a protective group show the lower means upregulation, downregulation of the MTB infection gene, and also the downregulation of most of the immune gene in the TB infected mice. Okay, so as conclusion, I hope I'm on time. As a conclusion, the ethyl acetate extract from the Streptococcus leaf exhibit a robust potential for immunomodulation activity in a treatment group. And the ethyl acetate extract of Streptococcus leaf also demonstrate protective against the mycobacterial tuberculosis in protective group. So this suggests that presence of potential active compound with anti-TB activity in CTL extract. So, and then maybe after this, we will further study to see what is the compound responsible for this activity. Yeah? So eventually, of course, we want to develop maybe a drugs or new neurosteroidal product that can treat or inhibit the TB. Okay, last but not least, we also studied on the anti-dengue activity. So here we use the protease inhibition assay. So I think this one, Pak Cahyo more expert than me about how to sequence the protein whatsoever to purify the protein and so on. This is the process. At the end, we get the purified protein, which is here is the NS2B, NS3 Pro, which is the protein that is responsible for the virus replication. And then this is how we do the assay. So we use here is the concentration of our sample is from 12.5 to 400 microgram per mil. We use this substrate for this assay, and also and after that we just put in a fluorescent spectrophotometer and then measure the IC50 value. Okay, as a result, we can see here for this. I apologize because we just only do the two type of solvent here, which is the aqueous and ethanol. We still not managed to do the ethyl acetate and hexane extraction. But preliminary study done show that the ethyl acetate and hexane is quite low. The IC50 is quite high. That means the anti-dengue activity is not that high. So maybe we can conclude here is in all the 52 sample, the ethanol extract have the highest anti-dengue activity. For example, here is the RAL is Rhizophora apicutulata leaf. This one is leaf, have the lowest IC50 value, which shows that it's the highest anti-dengue activity from these all 52 samples. Okay, so this one we just propose how it works, how the mangrove works in anti-dengue activity. So what they do maybe is just they bound with the active site of the enzyme here and they block the substrate from combined with the enzyme. That's why we see the inhibition occurs here. Okay. our student also do the correlation with the phenolic content in the student, even though the highest anti-dengue activity, the sample that have higher is not have the highest total phenolic content or antioxidant, but their total phenolic content and antioxidant is among, I think, top 10 high. So we can say that maybe the anti-dengue activity in this sample is maybe part of it is contributed by these phenolic compounds or antioxidant compounds. Okay, this one just for, and then just for comparison with the other studies, our studies actually have a lower ICC value. That means our study shows that this mangrove extract have quite high anti-dengue activity compared to other studies. Okay, we do some LCMS study just to see what compound inside of this, the highest sample for anti-dengue. So we found that this is the 4 main compounds inside of the Rhizophora apicauda leaves, or 5, sorry. So when we search about this compound, 2 of the compounds is already reported to have the anti-dengue activity. And then 1 is hesperidine, also have an antiviral against the SARS-CoV-19. And then chlorogenic acid have the antimicrobial activity. So yeah, in future, of course, we will try to find which the real compound that contribute to the anti-dengue activity in this. Okay, as for conclusion, most of the mangrove extracts selected in the study possess anti-mycobacterial and anti-dengue activity. And future direction is, of course, we need to isolate, do the fractionation. A lot of things still need to do. And we hope that in future we can get maybe novel compounds, maybe, and also we can develop some products or some drugs that can inhibit TB and also maybe to treat dengue also. That's our hope in future. So as an innovation, so I want to thank all my colleagues in University Malaysia Sabah, especially Pak Chayo here, also helped me a lot with this study. Dr. Zarina, Dr. Atira, Tamar Kansil is my PhD students, just finished her viva last week. Ain is my master student, she's already graduated last year. And also thank to Pak Collis from Swiss German University. So Pak Collis helped me a lot in terms of giving advice about the species or the extraction method and so on. And also, last but not least, the Sulaman Wetland Sanctuary, which is they provide us the sample for the study. Okay, thank you. Hope not too long.
Thank you very much, Dr. Ruzaidi. It's very interesting lectures. So the point that we understand that if we want to collect the special secondary products, so we have to find these plants that have oxidative stress. So it's also my recent interest because with my student recently we start to explore the endophyte from the geothermal plants. So it's very interesting. So yes, so for the next lectures, Mardani that will give a lecture about this natural product extract as a potential treatment for Alzheimer's disease. So, okay, before that, Dr. Marzadi graduate from University Science Malaysia from bachelor, master, as well as PhD with the subject microbiology and then molecular biology. Such interest eliciting mechanism of NIF change in nitrogen fixation process by prospecting of microbe and study on aging and age-related disease using Drosophila melanogaster. So the publication until now 21 published article, which is index 5. So without further ado, please, Dr. Mardhani, time is yours.
Sebentar ya, mau share screen. Assalamualaikum, selamat pagi. My name is Mardhani. Four of us from ITB UMS. So basically, I joined UMS 2 years ago. Without I'm telling you guys this, you also can realize that I'm much younger than Pak Cahyo. That's why I'm just joined UMS. Yeah. So basically, my interest is when I graduate for my postgraduate study, I'm interested in studying gene regulation of bacteria. After I graduated from my PhD, my interest is more towards studying human aging and age-related diseases. So for today, I'm going to share with you one of my projects during my postdoctoral study. So, bolehkah saya share mix bahasa sama English supaya memudahkan pemahaman ya?
Yes, of course. Yeah.
So this presentation is too technical in terms of biology punya term semua kan. I'm afraid that most of you not familiar with it, so I try to explain it as layman as possible. Okay, next. So pertamanya, what is aging? Aging itu apa? In general, by terminology, aging is generally defined as progressively getting older, and then it is coupled by senescence. So this is the terminology. If you want to look at the situation, you can compare me with Pak Cahyo again. So I'm younger, so as I get older, I will become like Pak Cahyo. So that process is called aging. Aging process. So next. Yeah, this can be depicted, boleh ditunjukkan di dalam gambar ini. So aging is a process of getting older. Okay. Once we get older, apabila kita semakin tua, maka datanglah pelbagai penyakit. Dan salah satu daripadanya adalah, kalau di Malaysia kan kita ada itu penyakit 3 serangkai, ya. Darah tinggi, kencing manis dan jantung. So itu adalah common penyakit tua. So saya melihat di sebelah lainnya, yaitu Alzheimer disease. kalau bahasa biasanya dipanggil nyanyo. Ya, nyanyo. Sini apa ya? Tapi itulah Alzheimer disease, pelupa, dia jadi pelupa. Ya. So, siapa yang menemukan Alzheimer disease ini? Inilah bapaknya, Alois Alzheimer. So, kalau ibu-ibu bapa semua menemukan satu penyakit baru, boleh dinamakan dengan nama ibu-ibu dan bapa-bapa semua. So, penyakit ini ditemukan di sekitar awal tahun 1900, di mana pesakit itu mengalami pelupa, tidak ingat semua. Kemudian brain autopsy dijalankan dan mereka melihat otaknya itu mengecil, mengecut. So, jadi keadaan ini penyakit ini dinamakan sempena dengan penemunya, iaitu Alois Alzheimer. Sampai sekarang kita gunakan Alzheimer disease. Ini adalah gambar raja di mana di sebelah kirinya otak manusia yang normal. Di sebelah kanan adalah otak manusia yang menghidap Alzheimer. Alzheimer disease ini dia disebabkan oleh aggregation of this protein called beta amyloid. Beta amyloid adalah sejenis protein. Apabila kita semakin tua, dia tidak dapat diproses, dibuang dengan cara yang betul, maka dia akan melekat di otak kita. Secara specifically ialah neuron, dia akan mematikan neuron itu. Apabila lama-kelamaan neuron akan mati, komunikasi antara neuron itu terhalang. Jadi apabila terhalang, kita lupa. Itulah bermulanya penyakit Alzheimer disease. Puncanya ada pelbagai. Salah satunya adalah oxidative stress, mitochondrial dysfunction. Ini adalah terminologi yang mungkin ibu-ibu bapa-bapa kalau boleh faham, bagus. Kalau tidak, saya just go through sajalah. Dan cytotoxicity, di mana toxic produced by this beta amyloid protein. Kemudian lama-kelamaan otak itu akan mengecil. Dan eventually dia akan berpindah kepada demensia. Demensia ini adalah Alzheimer adalah salah satu daripada demensia. Demensia ni dia ada banyak jenis lah. Ini lebih teknikal lah. Salah satu puncanya adalah disebabkan oleh mitochondrial dysfunction, di mana amyloid beta peptide ini dia deposited into the brain. So kalau ibu-ibu bapa-bapa boleh lihat, dia akan deposited di kawasan ini. Dia akan terkumpul di situ, menyebabkan sel-sel neuron mati. So lama-kelamaan dia akan jadi otak mengecut lah situ. So that basically is the summarize of mitochondrial dysfunction. Yang kemudiannya adalah cytotoxicity, seperti yang saya bagi tahu tadi. Ia adalah toxicity yang disebabkan oleh di housekeeping of that amyloid beta protein tu. Dia tidak dapat dicernakan, dibuang dari badan dengan cara yang betul. So apabila dia tidak boleh melalui jalan yang betul, dia akan melalui lagi satu pathway yang menyebabkan beta amyloid tu menjadi toksik kepada brain cell kita. Jadi ini adalah satu lagi gambar raja di mana brain atrophy terjadi akibat daripada penyakit Alzheimer disease ini. Kalau dilihat di sini, dia situasi ini dipanggil atrofi, dia mengecut berbanding dengan sebelah kanan yang normal. So kalau dilihat sini juga kita nampak macam ada hollow di tengah brain itu akibat daripada deposition of the amyloid beta. Jadi amyloid beta ini, kalau kita lihat neuron ini, neuron adalah sel otak kita. Amyloid beta dia akan membentuk plak deposited di kawasan neuron sini. So dia eventually dia akan menghalang the function of the neuron itu sendiri. So dia akan melekat di situ. Nah, ini lagi satu gambar raja menunjukkan neuron di mana amyloid beta plak akan terkumpul di sekitar kawasan ini. Kemudiannya ada lagi satu tau protein, dia dipanggil tau protein, adalah salah satu punca juga di mana neuron itu akan mati. So, introduction to amyloid beta, dia seperti yang saya beritahu tadi, the aggregation of beta amyloid, you won't realize it until it is too late. Dia tiada simptom, tiada apa. So even now, maybe the amyloid beta is aggregate into your brain already, tanpa perlu menunggu kita tua. So the process is really not really well understood. We have late onset Alzheimer disease or early onset. Early maksudnya kita sekarang ini. Mungkin proses ini sudah berlaku, tapi tiada biomarker untuk kita tahu adakah benda itu sudah terjadi ataupun tidak. Macam mana, bagaimana amyloid beta protein ini menjadi toksik kepada otak kita? Kalau dilihat on your left hand side, it is non-amyloidogenic pathway, di mana non-toxic pathway secara ringkasnya. Ini adalah amyloid precursor protein. Okay. Kalau di bahagian yang di pathway yang tidak toksik, alpha secretase gamma secretase akan memotong sini dan dia akan riliskan P3 protein dan AICD, which is non-toxic. Jadi ini adalah normal pathway di mana kita membuang, istilah membuang this amyloid precursor protein. Apa jadi kalau dia tidak melalui jalan non-toxic? Kalau dilihat di sebelah kanan, we call it amyloidogenic pathway, which is a toxic pathway. Di sini, beta secretase akan cleave the APP protein here and produce this amyloid beta. Later on, it will be cleave again to produce this amyloid beta saja. This amyloid beta will form oligomerization. Dia akan berkumpul banyak. Eventually, dia akan form plaque. So this plaque will be deposited into your brain, and dia akan bunuh the neuron. So that is the process of how the Alzheimer disease terjadi. Ini adalah gambar rajah beta amyloid. Kita ada dua. One is beta amyloid 40, AB40. Another one is AB42. AB42 dia ada extra 2 amino acid, isoleucine and alanine here, which is more toxic compared to beta amyloid. This table here showed that the genes involved, the protein involved in the Alzheimer disease, in the oligomerization of the beta amyloid. So I'm not going to go through this very deep. These are the list gene-gene yang protein yang involved dalam proses pembentukan beta amyloid itu sendiri. Okay, jadi kita sudah tahu apa itu Alzheimer disease. Bagaimana kita hendak study the disease itself. So in biology, we have a lots of model yang boleh digunakan untuk mengkaji human and human related disease. Seperti Pak Ruzaidi guna model tikus semata tadi ya. So kita ada C. elegans, sejenis cacing. Ibu bapa biasakah dengar C. elegans? Cacing yang sangat kecil. Jadi dalam presentation ini, I will use Drosophila melanogaster. Kalau di Malaysia kita panggil dia lalat buah, sama di sini. So I will use this model to study Alzheimer disease. So, bagaimana? Okay, next. Ini adalah comparison antara model-model yang kita gunakan untuk mengkaji human and human related diseases. Kalau kita lihat di sini, apa yang keistimewaan Drosophila ini? Nombor satu, progeninya sangat banyak. Dia boleh menghasilkan banyak anak. So, kita not really difficult to get the samples. Kemudian maintenance-nya juga sangat murah, tidak begitu mahal. Dan paling penting adalah kita boleh menggunakan genetic tools untuk melihat mutasi-mutasi jin supaya kita lebih memahami penyakit tersebut. Itu yang paling penting. Ketiga-tiga ini kita boleh mengubah jin-jinnya, tetapi untuk tikus jangka hayatnya adalah sangat lama. Oleh itu, tidak sesuai untuk kita gunakan sebagai screening model untuk mengkaji penyakit manusia. Untuk C. elegans, dia tidak mempunyai karakteristik fenotipik, kita panggil kalau di biologi, phenotypic changes yang kita boleh lihat perubahan sekiranya cacing itu ada Alzheimer disease. Apa yang kita boleh lihat di situ? Jadi menggunakan Drosophila sebagai model, kita boleh menginducekan Drosophila itu untuk ada penyakit Alzheimer. In general, Drosophila itu sendiri tidak ada gene amyloid beta. Dia tidak akan dapat Alzheimer disease. Tapi kita memaksa Drosophila itu disebabkan kebolehan kita untuk genetically manipulate the Drosophila. Kita paksa Drosophila itu untuk menghidap penyakit Alzheimer. Okay, next. Ini adalah why we choose Drosophila. Is the most important thing is genetically amenable. We can play around with the genetics untuk tengok the disease and so on so forth. And then 50% of fly genes having homolog. Maknanya gen-gen yang kita ada 50% ada di Drosophila. Dan more than 70% of human disease can be found in Drosophila also. Maknanya kita ada penyakit jantung, kita boleh tengok di Drosophila, Alzheimer, metabolic disorder, semuanya kita boleh modelkan di Drosophila itu sendiri. Drosophila research ini telah bermula di sekitar tahun 1900. Bapanya adalah Thomas Morgan. So dari situ penyelidikan Drosophila berkembang sehinggalah ke hari ini. This is just the history of Drosophila research. Drosophila melanogaster, the genome size is very small, around 175 megabase. Fahamkah ini term-term ini? Di dalam the genome maksudnya the DNA yang macam mana? Keseluruhan jinnya. Ya, the genome. Ya, maaf ya kalau tidak paham, sebab the term is very specific. Dia ada 4 kromosom di sini. Salah satu daripadanya adalah sex kromosom. Jadi untuk female dia akan ada X, untuk male dia akan ada Y. So these are the the genome and average lifespan, maksudnya purata kehidupan Drosophila ini adalah sekitar 60 hari saja. The life cycle is very short. That's why we can use this as a screening model. Okay, this is the life cycle dari bertelurnya Drosophila itu sehingga dia hatch sampai membesar adalah sekitar hari 12. Bermula dari situ dia ada period of 60 days approximately untuk hidup sampai dia mati. And most important thing is the female become sexually matured selepas 8 ke 10 jam selepas dia keluar dari pupae. So it is a very fast process. Okay. Drosophila as a model. Bagaimana kita secara umumnya Drosophila itu sendiri tidak mempunyai penyakit Alzheimer. Bagaimana kita mahu force the Drosophila to have the disease? So we use human amyloid beta gene yang seperti saya beritahu tadi. That gene we inserted into Drosophila so that the Drosophila will have the disease. So selepas Drosophila itu menjadi Alzheimer disease, how are we going to treat? What are we going to do with it? So in this project, we use the natural product, which is danshan in Chinese, danshan, as a potential to treat the Alzheimer disease. The process is almost the same as Pak Zaidi tadi, di mana kita mengambil root extractnya, we have water extract and methanol extract. Dalam danshan itu sendiri, there are two very famous compound we call it. Danshan 1, danshanase. Dan kemudian yang keduanya adalah salvionolic acid. The derivative of salvionolic acid, dia ada sal A, B, C, D, E, F, G. Why I choose this danshan as a drug to treat Alzheimer disease? Number one, sal A and sal B, they have very high antioxidative activity. Sal A was able to decrease metal-induced amyloid beta aggregation in previous study. And sal B showed selective inhibitory against BAC1. BAC1 is one of the genes involved in the production of amyloid beta. With this study, they do it in vitro. Now what I'm doing is to quit in vivo, to feed the Drosophila with this extract and see the effect of it. So how are we going to do it? Kalau lihat sini, this is the vials where we grow the Drosophila. Ini semua adalah Drosophila. Dan di sini adalah makanannya. So the way we do it is we put the drugs ke dalam makanan ini. So the Drosophila will feed, dia akan makan. Itu, and we will look at the effect later on. So there are 3 types of analysis. 3 jenis analisis yang kita mau tengok kesan daripada that drug treatment itu. Nombor 1 is neurotoxicity assessment, which is we're going to look into the eyes of Drosophila. Later on, I will explain it. Kemudian kita akan lihat berapa lama dia boleh hidup. So, in general, people who get Alzheimer disease, in general, dia akan mati cepat lah dalam bahasa kasarnya. Okay? Kita nak lihat berapa lama dia boleh hidup before and after the treatment. Dan kemudian, kalau seseorang itu menghidap Alzheimer disease, they will tend to have movement, locomotive impairment. Mereka susah bergerak. That kind of traits we're going to analyze using Drosophila. So we're going to use 3 types of flies. I'm sorry if this is too technical. Okay, next. Bagaimana kita mahu lihat the expression of the amyloid beta? Fahamkah apa itu expression, gene expression? Kita mau paksa Drosophila itu menghidapi Alzheimer disease. We use this system, which is called GAL4 and UAS expression system. Sistem ini diambil dari yeast sebenarnya. So because of the genetically amenable of the Drosophila, we can use this yeast system in the Drosophila to express every genes. Kalau lihat di sini, DMR means glass multiple. It is specific di matanya, the promoter. Ini adalah promoter. Familiar with protein promoter? Promoter ini dia akan express the GAL4 protein. The GAL4 protein akan bind kemudian this will express target protein amyloid beta. So if we are not studying amyloid beta, for example, we studying other diseases, we can force the disease to have it in any part of the Drosophila untuk lihat the phenotypic changes, not necessarily in the eyes. For this study, Alzheimer disease, I express it in the eyes. Maksudnya, penyakit Alzheimer itu tertumpu di mata lalat itu. Dia tidak ada di bahagian lain. This is because the tissue specific promoter that I use. If I use different promoter, for example, I'm also studying testis, Drosophila testis. If I change this to testis promoter, the Alzheimer disease will accumulate di dalam testis Drosophila itu. Jadi di sini amyloid beta kita boleh tukar ke different disease. This is just US Galpo system work. Okay. We cross them together. Cross means kita masukkan jantan dan betina. Cross, they will have F1. So the F1 will express the amyloid beta protein. So di anak-anaknya akan menghidap penyakit Alzheimer. Another driver that I use is actin galpo. Tadi di GMR itu dia tertumpu di mata. Actin galpo dia ada di keseluruhan lalat buah itu. Jadi protein yang diekspres dalam ini adalah GFP, green fluorescent protein. Kemudian kita crossbreed, the F1 will show fluoresce, the green fluorescent protein. Dia akan express di keseluruhan badan. So this is how we play with the genetic of expression. Okay. Apabila kita induce the Drosophila to have Alzheimer disease using GMR, glass multiple reporter, you can see here di sebelah kiri A is wild type, normal, tiada penyakit. If we overexpress it di sebelah kanan B, you can see ada lekukan di matanya. Dia menjadi tidak cantik in general. That is how we visualize the Alzheimer disease splice. Call it as rough eye phenotype. This is the wild type, looks very beautiful, the hexagonal shape, and this is totally disrupted eyes due to the overexpression of beta. Okay, now using my drug Danshan, we have Danshan water extract, Sal A and Sal B that we use to treat the blood. The circle here showed the rough eye phenotype. Maksudnya di sini, the lekukan itu tertumpu di kawasan itu. We want to see the how much that we need to treat, minimum macam MIC, amount of drug that we can use to see the phenotypic changes. So we look at the phenotypic changes. How are we going to quantify? Bagaimana kita mahu tahu berapa banyak the recovery process? So we use a software called Phenotyper. The fundamental of it is dia akan measure the perfect hexagonal shape ini. So for wild type control, you can have perfect hexagonal. Then they translate it into P score. For subtle rough eye, very rough, kalau tengok sini, the very rough one, the trajectory is totally lari, it's not hexagonal shape anymore. This is how we quantify the degree of severeness of AD fly, Alzheimer's disease. So if you can see here, this is negative control. The score is 1. We normalize it to 1. This is wild type, sorry. We normalize it to 1. So the degree of damage is very high, you see here. And then Danshan water extract. Anything that goes lower to the untreated one, we consider it as they have the recovery effect of the drug. The Dunshun water extract, most. So in this case, all three drugs that derivative that we use give positive effect to the flies. And you can see here, this is the SEM, scanning electron microscope. The wild type one look very nice. This is the untreated. Bottom C is Dunshun water extract. D is Sal A and E is Sal B. So we quantify it using the phenotyper just now. So, as I said just now, if the flies have that Alzheimer's disease, they will live relatively shorter than the wild type one. So this is the longevity analysis that we perform. Of course, the treated and the wild type shows longevity, live longer compared to we can see that females live longer. But this is the result that we get. Next. Okay. For movement, geotaxis, how are we going to look? Macam mana pergerakan lalat itu? We're going to analyze it using geotaxis assay. If you look at from the left is the treated flies, the middle one is untreated, the right hand side is control. Kalau kita lihat sini kan, the control one positive with wild type one, movementnya lebih pantas. Please, dia bergerak sangat pantas ke atas. Cepat ya. Maaf, sini kena cepat. Oh, kami di Malaysia pantas itu cepat.
Very fast.
You want to see it again?
Yeah. Okay.
Okay. Again, the left hand side is treated. You compare with the middle one, untreated. Dia naik, dia jatuh. A few. Now look at this one. So, dia jatuh. So, the untreated one, dia ada satu phenotype yang dia tidak larat, panjat-panjat jatuh, dia lemah. Compared to the treated one. Right? The control non-Alzheimer disease one, of course, dia akan terus bergerak ke atas. This is how we analyze the movement effect of the drug that we use to treat the Alzheimer disease. The rescue effect, we call it. This is just to show, I summarized it in the video just now. So this is basically the numbers that we have from the end. Then, okay. Selepas lalat itu ada Alzheimer disease, we do all the previous analysis. We want to look deeper into the gene regulation of the flies. Apa yang terjadi di dalam itu? So we did transcriptomic analysis. Faham? Familiar with transcriptomic? Yeah, we did transcriptomic, melihat gene regulation dalam tu, the global gene regulation. So we have the treated one and the untreated one as a control. Kemudian kita bezakan di mana gene, kalau Pak Zaidi tadi dia gunakan qPCR, which is smaller scale compared to transcriptome. Transcriptome dia melihat kepada keseluruhan lalat itu. Apa yang terjadi di dalam itu selepas kita give the drugs to the flies. So this is we call it enrichment study, enrichment of pathway. Di dalam pathway protein coding gene ini semua, dia tertumpu di situ. Itu yang banyak kita lihat terjadi. nombor-nombor ini adalah negatif, ini bermaksud down regulated. Kalau positif atau tiada tiada tanda negatif, that means it is up regulated. So this is just to show you what type of analysis that we do. We look at down into the molecular level of Alzheimer's disease itself. Okay. This is another example. Just now we have cell A, now we have cell B. Try to really understand the mechanism of the drug, how the drug rescued the Drosophila. And then itu adalah untuk Alzheimer's disease fly. Apa yang terjadi kalau lalat itu menua secara semula jadi tanpa ada Alzheimer's disease? So we want to look at the global gene expression again. What actually happened? Can we map out the pathway yang Alzheimer's disease flies itu ada dengan yang naturally get old flies tu? Kita mau melihat ada kesinambungan atau tidak. Apa yang terjadi dalam tu? Again, we did a transcriptomic studies. Oh no, we map out dulu. Kita lihat dulu berapa lama lalat itu boleh hidup. So in this study, they can live approximately up to 70 days. It depends on lab condition. Sometimes 60. Depend on lab condition and the food that we give it to them. So in my study, we have up to 70 days. So we are going to look into the gene regulation pada usia mudanya, pada usia remaja, kemudian tuanya. We try to mimic human aging process. Okay. This is very technical. This is how we did the transcriptome. We have, like I said just now, we have day 1, day 30, and day 60 compared to human aging process. Kalau day 60 tu dia sudah lebih kurang Pak Cahyo lah, sudah tua tu. Muda dek, oh betul. So how we do it? To study the gene expression, we need to extract the RNA. So the RNA that we have, it will be processed and we will sequence it. Penjujukan RNA, kalau bahasanya. So selepas penjujukan, kita akan dapat raw data. This is very technical. Maaf saya explain truly saja. Kita dapat kita proses di raw rates. Kemudian kita gunalah segala software yang ada di sini supaya analisis yang kita dapat adalah gene apa yang terlibat, gene apa yang up regulated or down regulated. This is very macam bahasa kampung to explain this very technical stuff. Tapi dia bukan jangan macam mana? Don't it is not the correct term to use it, but I'm using it to explain so that semua dapat faham lah basically. But again, this is the heat map showing berapa banyak yang up regulated, berapa banyak yang down regulated. So what is up regulated, what is down regulated? Kalau biasa nombor itu adalah kosong. Jika kita beri makan sesuatu kepada lalat itu, dia boleh jadi dia turun. The gene expression tu go down. Maknanya down means dia try to inhibit something. Kalau biasanya dia adalah contoh 5. 5 itu dia menghasilkan penyakit Alzheimer, for example. Dia perlukan 5 untuk hasilkan penyakit Alzheimer. Jika down regulated bermaksud dia hanya ada 3. So kalau 3 itu dia sedikit sebanyak akan inhibit the Alzheimer process. For up regulated contoh 9. Maka Alzheimer disease tu lagi banyak lah. That is just layman to explain what is up and down. So we have the numbers here. Setelah kita buat, we compare yang tua dengan yang remaja, yang tua dengan yang muda, remaja dengan tua. We do pairwise comparison untuk lihat apa yang terjadi dalam proses penuaan itu sendiri. These are the genes that we have the pathway, the enrichment pathway that we have. Kalau lihat sini, the upregulated lebih kepada histone modification, which is epigenetics. Yalah, bila tua macam-macam akan jadi kan. So that shows the aging process itself lah, basically. So I'm not going to details of this thing. Basically, after we do the transcriptome, this is the result that we got. We know overall what actually happening. Then from the enrichment itu, the genes that we have to, we map into human Alzheimer disease pathway. So these are the pathway for human Alzheimer disease. Because earlier I said they have ontology. Lalat itu boleh dimashkan kepada manusia, genes of human. So this is what we have. The green one, I'm not going to do this, just to show you, yes, we can map to humans pathway, and we can identify which genes that involved in this and that process. For example, we use the drugs, and then we can see certain gene here is acting weirdly. In normal condition, they are up, for example. So after we treat, we see that this gene down. So this gene must heavily involve selepas kita beri drug itu. So this gene can act as central untuk kita map up the pathway from the drug that we use, the mechanism of action of the drugs. This is another example. This is more to longevity pathway of human. The same also, we can see they are green, they are red, showing the regulation of the genes. This is to summarize what really happened when we do the transcriptome. I'm not going one by one, just briefly show you. These are the person that involved throughout the experiment. Actually, I did it when I was a postdoc in USM. My team, these are the PI here. I'm just the lab itu. Yeah. Okay, before I end my talk, I would like to share what actually in IPB, what actually we have. We have genomic library. We have high-end equipment, for example. This is the sequencing machine. Yang we did the transcriptome, we can do it here. We have the machine in-house. And from the machine that we have, we managed to secure lots of grants, and this in microbiology lab, and we managed to secure huge grant also. I'm just showing the numbers so that ibu-ibu bapa-bapa sana tu kalau berminat nampak banyak duit kan, datanglah di UMS untuk study kan. Selalunya banyak duit, senang buat research kan. So I think that's all from me. Thank you. If you have any question, I will be happy to test this.
Okay, thank you very much, Dr. Madani. It's a very interesting lectures. It's bring my memory when back to my master when I still work with this. So maybe in this room only me and Pasari that can completely understand your lecture, but it's yeah. We can bring this topic to our lectures to develop the understanding about in this area that the developing in this genetic molecular study is very enormous. Okay, thank you very much. Okay, for the last, Dr. Khairul Asfar that will present about towards the development of next generation nano vaccine. So this very interesting nano vaccine, utilization of peptides and oligonucleotide. So Dr. Khairul Aswar, graduate from University of Queensland. Without further ado, please, Dr. Khairul's time is yours. Yes, of course. Yes, yes, yes.
Hello. Wow. So, halo, assalamualaikum dan selamat siang rakan-rakan sekalian. My name is Khairul Asfar. My background actually is in medicinal chemistry, but that was when I was studying. But since entering into University Malaysia Sabah, I have dwelled in a lot more other things, such as metabolomics, natural products, and so on and so forth. One of the things that I was planned to do that I unfortunately could not involve in my presentation is to use a research whereby I'm using black ginger to improve sexual performance. Yeah. Tapi unfortunately, it doesn't fit into the theme of our presentation. So hopefully I can be invited again next year to share on that work, and hopefully the theme, sustainable development. But for today, if you allow me, I would like to share a little bit about something to do with nanovaccines, whereby since the happening of COVID-19, there has been a lot of focus towards developing vaccines, and specifically in nanovaccines. So although I might not be the foremost experts in vaccine, hello. Okay. But there are a few things that I am focusing on, and that's, I think I can safely say that I might be one of the few in Malaysia who is adopting these types of strategies. So hopefully you'll stay tuned until the end. So fasten your seatbelts and let's begin. Okay. If you've noticed, vaccines previously is always focused on preventing certain types of diseases, right? However, currently the focus of vaccines has started towards preventing the disease, but actually you would have to have the capacity towards treating the diseases first, while in the future also in preventing that particular types of diseases. So if you look at throughout our history, there has been a lot of vaccines already on the market, already being used in pharmacies, which bukan di sini dinamakan seperti apotek dan sebagainya. So there are a few things that probably you've already taken in your adolescence: TB, BCG, rubella, polio, and so on and so forth. However, since the happening of the COVID-19, then we've had a lot of vaccinations occurring that we had to take in order to adhere to probably the national immunization program to prevent the spread of the COVID-19 illnesses, right? So there has been a lot of that, such as AstraZeneca, Pfizer, and so on and so forth. Sama di Indonesia juga? Yes? Okay. So before I go into specifically on the body of work, I would have to just a quick introduction, whereby this is a compilation of various different strategies towards trying to treat different types of diseases. So the diseases are very distinct between one and the other. So hopefully we'll be able to follow. Yeah. So one of the first work is involving using a conserved region from a Streptococcus pyogenes, or also that can elicit group A Streptococcus diseases. So what we do, this is a work when I was in my studies, which Unfortunately, I'm still ongoing and helping out with this after I've already worked. In that we are trying to develop multi-strain vaccines to treat group A streptococcus. If you don't know what group A streptococcus is, that it is something seperti cacar begitu, cacar, yeah. So it is a bacteria that's able to infect you and causes a lot of issues with your respiratory diseases, as well as forming a lot of skin issues. And if left untreated, will drag and prolonged into something like a rheumatic heart disease or scarlet fever. So when that happens, unfortunately, the survival rate is unfortunately not too high. So currently there has been about 1.8 million cases globally, and roughly around 500,000 deaths. Not so bad lah kan, mungkin jatuh kan. For your information, Streptococcus pyogenes has a lot of strain, whereby unfortunately group A strep falls under one of the most deadliest strains of the Streptococcus. And from then on, it can lead on, like I mentioned, to something like the rheumatic heart disease and so on and so forth. Now, the strategy towards tackling this disease is to use the M protein, a specific protein from the bacteria, which is the M protein, a major virulence factor that is causing all the illnesses and the infections from that one particular segment of the bacteria. For your information, that group A streptococcus is a gram-positive and can be treated with antibiotic. But since its first discovery in roughly around 60 to 70 years ago, it has mutated into a antibacteria resistant strain. So current prophylactic strategies are still not able to treat that. So the best strategy moving forward is to use vaccines. Now, from the M protein, because like I mentioned, it has various different strains of strains. So from the M protein, we tried to isolate and locate and identify a conserved region which is similar within all strains of the Streptococcus. And we have found that within the C2 region of the M protein. Now, from that section itself, we have identified a minimal B epitope, which can elicit some to significant amount of antibodies. And that is highlighted within the green sequence that you can see here. There. Kalau saya tunjuk, nampak ke tidak kan? So from that, the first peptide epitope that was identified was a whole section from the M protein, which is the P145. And when it's being used, it was still eliciting severe side effects, which was the antibodies was attacking the heart muscles, unfortunately. So it was deemed unsafe and not usable for any commercial vaccine strategies. From there, they isolated or identified a minimal B cell epitope, which is the green, which was deemed J14I. However, it lacked the alpha helical conformation to elicit any significant antibody response. However, through using the ingenuity of a few people, unfortunately that wasn't me, they used an alpha helix promoting sequence to promote the alpha helix, which was deemed J14. And yes, they did found significant antibody response. However, the antibody response was not very specific towards the minimal B cell epitope, but instead mostly to the alpha helical promoting sequence. So our strategy for this one is to use, can we use two minimal B cell epitope, the J14I, together to promote alpha helicity? And this is what I'm going to share with you. Now, in order for us to elicit a significant antibody response with all vaccines, we have to use an adjuvant. So for this one, we are using a synthesized adjuvant, or also known as lipid core peptide, LCP, which is specific to the toll-like receptor, which can bind specifically, as well as being recognized by the toll-like receptors, which will bring the antigens towards the antigen-presenting cells, mature, and produce significant antibodies. So if you notice, it is made up of a lipid core as well as 4 linkers. And for this particular vaccine candidate, we use 4 copies of peptide epitopes in order to elicit hopefully a high antibody response. So ada soalan lagi? Belum lagi? Okay, nantilah ya. So to look specifically at the chemical structure, you can see that it looks something a lot like that. Now, the peptide epitope, the DJ14I, was coupled covalently with the LCP using copper catalyzed click chemistry. And when we test this under CD spectra, it was forming alpha helical conformation, as you can see by the CD spectra, which is good, which is what we wanted in the first place. Now, in terms of in the in vivo immunological evaluation, what we did was that we subcutaneously immunized B10DR mice for different types of vaccine candidates. So the first one is the LCP DJ14I. So the next one is DJ14I, delivered with a complete French adjuvant, a very toxic adjuvant, which is unfortunately is not legal to be used in human beings. A J14I, the minimal B cell epitope, also delivered with the CFA, the complete French adjuvant. The J14, which I mentioned previously, which was flanked with the alpha helical promoting sequence as well as a control. Now, if you can see, there are 3 different specific groups, which are J14 specific antibodies, DJ14I specific antibodies, as well as the P145. Now, the P145 for me is the most interesting because it is a whole sequence from the AIM protein. So if the antibodies can be recognized when we do verify it with the P145, that which means that we can technically say that the antibodies delivered from our vaccine candidates are able to elicit a significant amount of response in order to neutralize the infection from the Streptococcus. Now, if you can see, unfortunately, bad news is the LCP, the one delivered with our adjuvant, is not eliciting any significant antibody at all. Technically memang langsung tiada. Macam kalau di sini kosong maksudnya apa macam mana? Kosong? Kosong. So, however, DJ14I delivered with a complete French adjuvant, which is the toxic adjuvant, is eliciting some form of antibody response. Similarly with the J14, which was flanked by the alpha helical promoting structure. This shows there is still some potential, but however, it looks like the delivery system is tidak berapa memberangsangkan yet. Tidak berapa memberangsangkan lagi. So with memberangsangkan boleh digunakan? Oh. Oh. Maaf. So it's not very promising yet. So what we propose in the future is to probably modify the synthesized adjuvant in order to elicit a more significant antibody response. However, if you look at the DJ14I specific antibodies, when we deliver using the adjuvant, it is very similar to the one that is being delivered together with the complete French adjuvant, which is the toxic adjuvant. So technically speaking, in terms of verifying it and identifying it towards the complete sequence of the M protein, maybe not yet, but at least it is forming antibodies very specific to what is technically required. So we're getting there, but not there yet, okay? So the summary is that the delivery of the DJ14I with the lipid core peptide is unfortunately not working yet. I emphasize on the word yet. And it requires a little bit more of a modification to elicit some form of significant response. So bear maybe about a couple more years, then probably we'll get it right. However, one of the things that probably I would like to highlight from this particular work is that it proves that a very short peptide group is able to be derived from the conserved region of a particular protein of a particular bacteria to elicit a form of significant antibody response. Now remember that. Now, if you go home today, if you don't remember anything that I say today, remember that a short peptide can elicit a significant antibody response. Wow, mashallah. Now this brings me to the next. This brings me to the next work that we focus in. So the strategy for that one particular work, we adopt some strategies in order for us to develop a vaccine to combat not only COVID-19 but all types of coronavirus. Now this is something a work which is very recent that's being done. That's being done together with a few entities from University Malaya as well as International IIUM, UIA, University International Antarabangsa Malaysia. So there's a lot of us on here. and it's still ongoing. Unfortunately, belum lagi. So like I mentioned before, this is not to focus on one specific coronavirus. Yes, coronavirus, the recent coronavirus, SARS-CoV-2 is the most recent and the most popular, I would have to say, because out of all these coronaviruses, COVID-19 has caused roughly around what, 600 million cases worldwide since its first identification in the year 2020. So 2020, 2023, 600 million. Whereas if you look at other types of coronavirus, such as the MERS, Middle East Respiratory Syndrome, as well as SARS, SARS is only eliciting about roughly around a few thousands. Now, if we extrapolate on that data, imagine if there is another coronavirus infection in the next, na'udzubillah lah, beberapa tahun, then you can try to imagine that the numbers might be more than 600 million. So technically, we would have to prepare from now to develop something which can be specific not only to that future coronavirus, but also to the previous and the past coronavirus, such as COVID-19, SARS and MERS, and so on and so forth. Okay. So that's the strategy, that's the hope, that's the dream. Yeah. So with looking at what has been done to prevent and to try to treat and to avoid COVID-19, there has been a lot of vaccines. Now you've got all the very generic one types like Pfizer's, or the attenuated viruses such as apa yang dari yang kita inject tu apa tu? Not AstraZeneca, Sinovac, yeah, betul. So those ones are attenuated, and there's very different types of vaccines that's being used worldwide to try to combat. And it seems to be working because we have had no very serious issues with COVID-19 in the year 2020 yet, 2023 yet. Okay. So however, the strategy with this ones is that we are looking at different various strains of coronavirus, all the way from SARS, MERS, as well as the more recent one, the COVID-19, all the alpha, the beta, the gamma, the delta together. So we're trying to look at the sequences of the very specific spike protein to identify a conserved region, which is identical in all of those strains. And from there, we need to identify a minimal B cell or minimal B epitope that can elicit a significant antibody response. And from our work, we have identified that conserved region within the S2 region of the spike protein. So it involves a lot of in silico work, which focuses on the alignment of all those sequences, identifying the conserved region. And from that, apparently a very narrow region of the spike protein for all strains, we identified a couple of sequences which can be probably, hopefully be a part of a vaccine candidate in the future for multiple strains coronavirus. So there's a lot of softwares that we use, such as the Coffee, ITASA, and GrowMax, to name a few. Now, out of all of that, we have identified at least at first 83, and from there we narrowed it down all towards roughly about around 8. So we have shortlisted 8, which we will be testing when we do in vivo, and I will be explaining that in due time. Okay, so when we look at the peptides, so the peptides, if you notice, is 15 amino acid long. They are significantly different from one to the other, and also the structures of them is also very distinct. So if you can see, the first two maybe looks something like a beta sheet, while the others looks roughly like a random coil peptides. And all those numbers and figures too, mungkin you don't have to worry about that yet. Don't have to worry about that yet. So the most important thing is that we've narrowed it down from 83 down up until 8. Now, what we are trying to elicit is maybe a B from the B cell epitope, some antibodies, a memory cell antibodies, which can be used as part to prevent as well as to treat issues pertaining to coronavirus infection. Now, we hope to bind specifically to the MHC class 2 and the protein that's being simulated there, it looks like that. So what we hope is that peptide is able to bind complementarily as well as efficiently and significantly within the peptide binding growth. Thus, it's able to elicit some antibody response. So the most important thing is that look at the red color. Now, a few examples out of the 8, just a few examples, just the 2. If you notice peptide 3 and peptide 4, so these are peptide 3 and peptide 4's examples. Now, if you look at the sequence, their peptides, the amino acid sequences is very similar between one and the other. Maybe the differences is just between a few lettered amino acids, if you can see there. However, the interactions of those particular peptides with the MAC class 2 complex both different interactions whereby the binding energy is significantly different, as well as the H-bonds distance is very different. As well, if you can see, the peptides itself has very different structures between one and another, although most of the amino acid sequences are very similar between those two. So this is the beauty of changing 1 or 2 amino acids. And when we do that, we'll try to do a molecular dynamic simulation between those peptides with the MAC class 2 complex. And based on that, is just to summarize, we'll be doing molecular dynamics. Now, if you look at this, technically speaking, is it the best ideal molecular dynamics analysis? Probably not. Probably not. But at least if we can see that the fluctuations between one and the other are very different between one peptide and the peptide. Unfortunately, peptide 3 is fluctuations is a little bit more than that of the peptide 8. And for now, it's only within the 50 nanoseconds punya range parameter that we're looking at. However, we have done roughly about 200 nanoseconds, but unfortunately, with these types of work, it's still ongoing. It does take a long time for one particular peptide for 200 nanoseconds. And because due to our limitation, which whereby we are only using softwares which are free, so mungkin satu peptide tu mengambil masa berbulan-bulan. So unfortunately, it's still ongoing. But if you're looking at 50 nanoseconds, it's roughly similar between all 8 peptides. It's fluctuating between 0.1, 0.2. And thus, I think these are the peptides which we will be using for future in vivo works. Now, in concluding that in silico work, just right before I left for Indonesia, and we've already started immunizing the peptide candidates to biopsy mice. So what we hope to see is that very specific IgG antibodies that we can use to verify and link back to the spike protein. Okay, so that's that. So unfortunately, the mice are still waiting for me at home, and hopefully I survive the trip so that we can continue the work. So to summarize on this one particular work, in terms of in silico, we have identified a conserved region. And from that conserved region, a very small peptide, 15 amino acid peptide, has been synthesized to be tested via subcutaneous immunization to see whether or not the IgG will be able to elicit some response between multiple strains of coronavirus. Okay, so that's that. Hopefully, kalau saya dijemput lagi untuk tahun hadapan, mungkin saya boleh kongsikan yeah. Okay. Okay. So that's in terms of vaccines specifically. Now, the idea with that particular vaccines is that it is something like a preventive measure. Now, what I'm going to share next is something that which is also a preventive measure, but also to treat particular symptoms. So for instance, if you are ill with one particular disease, now these strategies will be able to treat that disease and prevent any future occurrences with those diseases. Okay, so what we're trying to do here is to focus more on gene therapy whereby we elicit or an extraction of proteins from the delivery of genes as a therapeutic response. So the focus here is on the delivery system itself. Now there are two various different delivery systems which I would like to share, which is the first one is peptide based and the second one is a lipid based. So the first one is involving a peptide. Now, gene therapy itself, in theory, that with all types of diseases, as long as you deliver an exogenous therapeutic gene, then your cells, your bodies will be able to express therapeutic response in order to treat those types of diseases. Now, firstly, there's various different strategies with gene therapy. I don't want to go into in-depth on it too much, but the first one is to replace the gene. The second one is to mask the gene. And the third one is to elicit a response or a protein response in order for your immune system to recognize those specific infected region and be able to treat it themselves. And there's a lot more there, especially since we've already discovered CRISPR, which is working to a certain extent. So the potential for gene therapy is limitless, numerous, and beneficial. Yep. Okay. However, delivering a gene in particular is very difficult, especially in order for us to traverse the cellular barriers involved with ourselves. So in terms of cell, you've got your cell membrane, you've got your endosomes, and you've got your nuclear membrane. So in terms of eliciting an expression, a protein expression, we have to ensure that your gene is directly delivered into the nucleus, so that the nucleus, the components within the nucleus will be able to elicit an expression, and in this case, a therapeutic expression. So there's a lot of barriers, some listed in these slides, but there are unfortunately a lot more that's been the bane of our issues in terms of eliciting a significant gene therapy. So one of the first delivery systems that's been used or has been investigated is the usages of dendrimers or polymers, a branching unit of polymers, dendrimers. Unfortunately, because due to the high excess of positive charge, then it is eliciting a very toxic effect to wherever that it's being delivered. So technically, it's not being used commercially as of yet. Thus brings why we are adopting peptides. Peptides is very small. Peptides has very lacking. If we need to incorporate positive charge, then the incorporations of those positive charge is very minute, thus negate the effect of toxicity, and as well it's highly customizable. We can probably synthesize it in the lab, and so on and so forth. So minta maaf rakan-rakan, saya lupa tengok korang juga. I have been always looking at them. So yeah, I'm sorry. But for this one, we have denote two very specific components for now. A very one involving a cell penetrating peptide to traverse through the cellular membrane, and the second one is as a dendron, a minor component of a miniature component from a dendrimer. So due to the reduction of the positive charge, hopefully it will not elicit any toxicity response. Now we are using L-lysine from that our body is naturally producing on a consistent basis as part of our synthesis, to ensure that this system is also biodegradable, as well as not an issue for future in the future moving forward. So the cell penetrating peptide that we're using is TET, derived from the HIV protein, but we are taking just a minor sequence from that to elicit the capacity for it for cellular uptake. So to have a clearer look at what the chemical structure of this system looks like is something similar like that, whereby the branching unit, the dendrons, are increasing from generations upon generation. So as the higher we increase the generation, the higher the positive charge. However, it is not to the level of the dendrimers, whereby the positive charge can reach between few hundreds to few thousands. But the ones that we're using is just as paling tinggi pun roughly around what 16. And for your information, the cell penetrating peptide itself also has positively charged amino acids in the form of arginines. So thus it's also contributing into the overall positive charge structure of the system. When we are looking at the capacity for it for cellular uptake, we can see that the higher the generation of the dendron, the better the cellular uptake capacity. However, it is still uncomparable to that of the dendrimers, which in this case, the control that we're using is about 25 kilodaltons of polyethylene imine, a branch polyethylene imine. But if you can see the significant between our positive control as well, the highest dendron generation is not too much yet, right? However, when we try to use this in order to elicit a response, so the cargo that we're using is plasmid, green fluorescence plasmid protein, whereby if it is successful, then we will be able to see expressions of green fluorescence protein. But from the ones that I've highlighted, out of all that darkness, you can only see 1 or 2 dots. And when we quantify that, unfortunately, it's only between about less than 1%. So can we say that this is a success, Probably not, because when it's compared to the positive control is eliciting roughly around 40 to 50% transfection. So 50% transfection in comparison to 1% transfection. I can safely say that this is a bust. Thus, what is the issue with that personally? Maybe our theory is that it's being entrapped within the endosome, and thus unable to be transported directly to the nucleus for efficient protein expression. So what we are trying to adopt here is to use a proton sponge mechanism, whereby at very acidic environment, there will be a component which is protonated and which will be able to burst the endosome, thereby allowing the the delivery system to travel from the endosome to the cytosol and into the nucleus itself. So for that, we are incorporating polyalhistidines, which is protonated at acidic conditions, which is technically very viable within the endosome. And we have synthesized various different structures of the systems in accordance, and to test whether or not, and to see which one is the most efficient. So if you can see, the dendron charge is still similar to the ones that I've showed previously, because technically the branching unit is still maintained, nothing so different. However, we have included either a linear polyalhistidine or a branch to see which one is efficient between all of this. So if you can look at the cellular uptake, now it has remarkable improvement in comparison with those without the polyalhistidines. However, in terms of the transfection efficiency, it is still also roughly just from 1, it raised up until 1.5. So is it still a bust? Probably it's still a bust, because like I mentioned before, the positive control is unfortunately roughly around between 40 to 50%. And the ones that we're eliciting for this one is roughly around 1 to 1%. So if you want to probably elicit transfection or protein expression in Drosophila, probably it might work, but not in human beings. And to have a clearer look, we tried to use a confocal microscopy to see exactly where these delivery systems are being located within the cell. So the ones that if you can see probably, so we've dyed each organelles very differently: blue for the nucleus, red for the endosome or lysosome, and the green specifically for the plasmid that we've delivered. And if you can see within the merge, probably you might not be able to see, but I'll just explain anyways, is that if it is merging between one with the plasmid is within the locality of a specific organelle, for instance, the nucleus, which is dyed in blue, then the color will change. But as much as we can see, probably it's only with changing color, not that much, and just very minutely in the changing colors from green to yellow. And these can be summarized as they are being entrapped, still being entrapped inside the endosome. So technically speaking, for this particular system, even with the inclusion of the proton sponge mechanism, it's unable to escape efficiently out of the endosome and not being able to be delivered directly to the nucleus. That's probably why we're having issues with to get significant amount of protein expression, unfortunately. So to summarize on that, peptide-based system is, I'd like to highlight that it's not a complete failure, because we are getting very high uptake, but maybe we need to tweak it a little bit more in order for us to play around with getting out of certain types of barriers. Okay, so although initially we tried to use this as the main delivery system, maybe we have to consider that it might not be suitable for the main component of the delivery system, but as a supporting unit of a different delivery system, which coincidentally that I'm going to share next. All right. So the next work that I'm going to share with you, just bear with me. This is the last one, I promise. Tidak lagi lepas ni. Is the development of a trilipid-based liposome. So we've heard about non-viral vectors previously. We've heard about things like peptide, like I just shared, but and also dendrimers and polymers. But the ones that I'm focusing here is to use something like a liposome, a lipid-based system. Now, our cell membrane is made out of a lipid chain. And if you look at your skin, no, you won't be able to see that lipid chain. But you can imagine that all of us, all our cells is being encased within our cellular membrane, which is made out of a lipid chain. So what we are trying to do is that with the formulation of these lipids, we're able to penetrate through that cellular lipid-based cellular membrane and able to overcome a lot of barriers that's proving an issue when we're using the peptide-based system. Okay. So, minta maaf sedikit, Lucha. So, firstly, the disease that we're focusing on is on breast cancer. Now, the breast cancer is very unforeign to a lot of us, and it's mostly, surprisingly, there are also cases in male, but the bulk majority of cases also occurs in females. Since the year 2008 to the year 2023, there has been a remarkably increase of cases for about 37%, whereby the last reported study to compile all the breast cancer cases is a bit roughly around 2 million. So globally, there's about 2 million people or more actually in the year 2023. Okay. So the current strategy towards treating breast cancer, unfortunately, is there's only two. It's you either go through via surgery or you either go through chemotherapy. Unfortunately, these strategies are very impactful, not only to the patient itself, but also to the surrounding friends, families, and the community. So what we're trying to adopt here is to use gene therapy in order for a more invasive treatment, so that the cells is treating the breast cancer, either the cells or the immune system is treating and combating the breast cancer issue from within and safely. Now, like I mentioned previously, that we are trying to focus on lipid-based delivery system, and there have been a lot of works focusing on lipid-based delivery systems. Now, what is the difference between the ones that I'm going to share with you now? For your information, that this is a brand new formulation, which utilizes 3 types of lipids all into one particular system. Now, the issue with that is that we have tried a lot of different types of library, and we found that these 3 works particularly well towards trying to develop a novel and effective delivery system. So one of them is using octadecylamine, which is technically mostly a positive charge, cholesterol, a neutral lipid, as a stabilizing agent, as well as DPPC, which is Zwitter ionic, which can compensate when the system is being delivered in various different pHs, such as acidic, basic, as well as neutral. Now, we've tried various different combinations, but we found that the best formulation, and the ones that I'm going to share, is just 2, DPPC, octadecylamine, as well as cholesterol, the 3-lipid system. And the second best is probably DPPC and octadecylamine. Now, we've tried, like I mentioned, I cannot emphasize this enough because there's a lot that we try through long hours. And for us to identify these 2 took us, saya ingat sampai kiamat, tapi alhamdulillah sebelum kiamat lah. So these 2 are the ones that is showing the best possible results that I'm going to share with you. So when we formulate those formulations, when we look at it under the microscope, it does form globules or something like a spherical formulations. And we hope that with these formulations, we are able to encapsulate whatever cargo that can be delivered to the breast cancer cell. So the ones that we are using it as a reporter gene is the PEGFP, also as a green fluorescence protein plasmid, which if successfully delivered and transfected will be able to elicit the production of green fluorescence protein, as previously that I showed before. Okay. Now, on a different note, that if you look at it via the TEM view, then you can probably see that there are structures within the encapsulation of the plasmid in comparison to that without any cargo delivered together. Okay. Now we have also evaluated various different ratios between the plasmid DNA as well as the liposome itself. When I mean that, when liposome, in that the concentrations of liposomes that we use is in ratio to that one particular unit of the plasmid DNA that we use. So we're trying to see whether or not increasing the concentrations of the liposomes are able to elicit any significant responses. Now, looking at the physical structure or physical characteristics of that, you can clearly see that the three lipid systems are able to compact nicely and more smallly, is that even a word, smallly, in comparison to that of two lipid system. So, yeah, penghazan. So, kena, teruskan saja. Okay, so if you compare between the zeta potential, they are both showing similar zeta potential of positive charge, whereby the DPPC octa and col are able to fully neutralize the charge at a ratio of 1.3 in comparison to that of the two lipid formulation, which is able to only neutralize this charge roughly between almost 1.5 ratios. So from this physical structure itself, we can see that the three lipid formulation is more favorable because it's forming very smaller nanoscale particles, which is ideal in order for us to get into through the cellular membrane, into the cell, and through the nuclear pore of the nucleus. And I have to probably emphasize that one of our strategies towards trying to get that inside the nucleus is via the passive transport through the nuclear pore, which requires foreign materials such as our formulations to need to be roughly around 200 nanometers in order for it to pass through via the nanopores. So although it might not be very ideal yet, it's roughly between 400 to 600 as of now, but the results that we found is a little bit surprising. Okay. Now this is, like I mentioned, the zeta potential were able to neutralize, and it was verified via gel shift assay, whereby the three lipid formulations is completely, almost completely at least, neutralizing the plasmid DNA charge at roughly around 1.3 ratio, and subsequently that of the two lipid formulation. Okay. Now, when we had a look at the toxicity profile of this, then you can probably see that, again, it might not be the most ideal because it's still showing some level of toxicity, roughly around about 70% of cell viability. And this is because of the bulk majority, or the bulk reason why it's toxic is due to the octadecylamine component, which is roughly around causing about 30 to 40% cell viability. So the bulk majority of toxicity, again, is driven by the octadecylamine component. In comparison between the three lipid formulation as well as the two lipid formulation, we can clearly see that the three lipid formulation is more, I won't say more safer, less toxic, less toxic than the two lipid formulation, because from the two lipid formulation, you're only seeing about 40 to 50% of cell viability, which is technically not acceptable in terms of an in vitro assay. Now, the best part about this work is that although, like I mentioned, we needed the size to be roughly around 200 nanometers, but the smallest that we got roughly was about slightly more than 400, less than about 500 nanometers, but we were still able to see protein expression from the green fluorescence protein of our assay. Now, although that the size, technically, what I want to highlight here, maybe size doesn't really matter. Tidakkah? Probably, because due to the bigger sizes of the formulations, we will still be able to get protein expression. And when we compare that to our positive control, which is a lipofactory LTX, we can clearly see that our formulations were producing a lot more green fluorescence protein than that of the physical control, the positive control, I'm sorry. And if you can see from the bright field that there are aspects which not really well presented from the toxicity that we see previously, because when I'm looking at the bright field, I don't really see any toxic effects on the cell itself. So this is something that we probably might have to revisit and to have a closer look. But the first initial responses to our work here seems that this novel lipid, especially the 3-lipid, the transaction that you're looking is a 3-lipid formulation, is performing exceptionally well in comparison to that of the positive control. So, yay. Okay. Now, our future prospects is that because we do have a lot of data on toxicity, on the size, that we would like to try to modify it a little bit more. We would like the size to be a lot more ideal, smaller probably. If you can get the smaller size, maybe we can get significantly more protein expression, as well as we would like to try to develop a very specific formulation which can clearly identify between disease cell and non-disease cells. Because with chemotherapy, like if you can consider chemotherapy, that the drug is affecting not only the disease area but also within the surrounding areas itself, and which is really severely impacting the livelihood of the patient itself. So what we're trying to do in the future is to develop a smart nanovaccine. Smart meaning small, multiple, alternative, responsive treatment. Ya kah? Tidaklah, mungkinlah. It's okay. You follow my paper later, ada tu. So what we're trying to do is to attach specific ligands, which is very specific to the receptors of, for instance, breast cancer cell. So those formulations will bind complementary on the cells of the cancer cells and would only act upon those cancer cells rather than all other normal cells. So that's for the future, probably. Hopefully, I can share those results if invited next year again. Kan insyaAllah. InsyaAllah. Kan. So I think that's all from me. Thank you very much. I would also like to thank a lot of people who have been involved on this particular work, from my alma mater, University of Queensland, from my current workplace, University of Malaysia Sabah, as well as all my friends from other local universities in Malaysia, such as UM, UKM, and IIUM. And these people have been detrimental, not detrimental. Yeah, UIA. So these people have contributed a lot towards the success of the works that I've shared with you. Some, although the work itself mungkin not yet a Nobel winning prize yet, but hopefully dengan doa-doa rakan-rakan sekalian mungkin bolehlah satu masa akan datang, kan? So that's all from me. Thank you very much for your time. And this is just a few of my friends and colleagues who have helped me all throughout the day, kan? So please ignore myself yang itu. Rambut dia, dulu saya jahil, so minta maaf lah. So thank you very much, and saya pas kembali kepada. Okay.
Thank you very much, Dr. Khairul. It's very enthusiastic and very generous sharing with us many information. I myself can follow the lecture from Dr. Mardani. But from Dr. Khairul, just technically, especially, is still okay. No problem. So it's only encouraged me to learn more. So, okay. So we have a hybrid class actually. So any question from the student? Okay. So first question. The other question, please prepare.
Okay, thank you for the opportunity. It's an incredible session, right? I'm very interesting about the percentage from Dr. Khairul. You have mentioned for liposome that that interesting for me is how can you make sure that liposomal drug formulation, the size is suitable for deliver to our body? Because the increase the size of liposomal when we absorb, it will depend, akan mendukung to support absorption in RES reticulum reticuloendothelial system, right? So how you can make sure the vaccine or the formulation it can suitable for the size, and then how to make sure the linkage kebocoran linkage for the formulation. Thank you.
That's very interesting question. So a few things that we have to ensure when we are doing this is one of it is, so I'll answer the second part first if you don't mind. So we would have to do a stability assay. So I did not share, but what we normally do with these assays is that when we do our formulations, we monitor it between day 0, from day 7, day 14, day 21, and so on and so forth. And from there, we are able to see that if the morphological structure of the formulation changes, then we'll probably know the most ideal time at which we have to probably deliver that. So for instance, that in most of our formulations, at least within the first 7 days, it's very intact, it's consistent, the size is consistent between the first 7 days. But from after 7 days, then it starts to aggregate, it starts to leak, like you rightfully mentioned, and so on and so forth. So we do monitor this via the dynamic light scattering, whereby we can look at the morphological of the lipids specifically. So for the first part of your question, how I think the ERS is in terms of to do with clearance, because there's a lot of issue pertaining to that. It won't be an issue if you're testing it in vitro, right? However, in vivo, like I mentioned previously, like towards the end of the my talk, is that we're trying to attach a very specific ligand that is complementary to the receptors on disease cells. So for breast cancer, there are various different types of receptors that's present in the breast cancer cell, but not present in other types of cells. So due from that, hopefully that the ligand are being able to recognize and being bind complementarily to those receptors and able to be uptaken directly into those cells specifically. But I can understand the issue, because sometimes you might say that what happens if I inject, if it's breast cancer, then but a lot of injection occurs in the shoulder and so on and so forth. So one of the strategies that a lot of researchers have tried to do is to probably introduce the formulations as close as possible to within the vicinity of the disease cells. And with that specificity and ligand and receptor, then there could be, unfortunately, we're unable to negate the effect of clearance completely, but at least we can ensure that the bulk majority of your formulation are able to be delivered into the specific target areas. So that's one way of looking at it. Yeah.
Cancer therapeutic is so toxicity, right? Okay, thank you, Doctor, for the answer.
Thank you. Another question? Yeah, monggo.
So you about the analysis itself, or you want to know about the machine? Or the application. So because okay, so the zeta potential. So zeta potential. All right. So with a lot of my work is mostly involving positive and negative charge, right? So with peptides, I was mostly highlighting on the positive charge. Similarly, with the liposomes, also there's an abundance of positive charge, because I'm using 3 different lipids, the positive, Zwitter ionic, which means that it can change between positive and negative, as well as a neutral charge. And these are very important because our cargo, the plasmid DNA, is mostly negatively charged. So we're trying to see whether or not the lipid is able to encapsulate the negatively charged. So the positive charge that we're using is bound or encompass the negative charge via electrostatic interactions, protect the negative charge, because if we deliver, as long as there's still negative charge of the DNA still exposed, then unfortunately when you deliver it inside your body, it will be cleaved or being dirosakkan. So what happens is that as long as there is a positive net charge, then it will be able to protect completely the cargo within inside, as well as it will be able to bind, not say complementarily, but bind to the surface of our cell membranes, because our cell membranes, surprisingly, is negatively in charge as well. So because due to the differences in charge, it can bind and release all the cargoes inside the cell membrane. So what we're trying to do is that we have to maintain, the zeta potential is trying to maintain that positive charge, but not too much. So because if there's too much, then you would probably see there's a lot of toxicity effects, but the ones that I showed probably is roughly around 40 to 50, which is still not ideal, because you can see there's still about 30% of toxicity. But again, I emphasize that there's still a lot of work that needs to be done. There needs to a lot of modification that needs to be done. So that's maybe the importance of the zeta potential that we're trying to do. Coagulation, yes, correct. So if there are still negative species within, so for instance, if within an environment there are still negative species, so imagine that your proteins also have negative charge. So it will be aggregating within our system, as well as in whatever environment that you have. Our worry is that rather than the lipid encompassing the plasmid is only attaching on the surface of the liposome itself. But from our preliminary work, we're able to check, and from our TEM, we're able to see that the cargo itself is within the liposome. And from our stability work, like I mentioned before, for 7 days at least, each formulation is very consistent, very stable. We still get particles of a good size, at least for the first 7 days. So what we can probably summarize from that is that if we need to deliver a formulation, or we need to elicit a therapeutic response, it has to be within the first 7 days on the current formulation before any modification. So the first 7 days is very crucial for us to elicit any therapeutic response at least. Hopefully, did that help? Boleh kah? Boleh. Okay.
Thank you. Another question? Okay, actually we have online, but until now there is no question. Okay, I have a question for Dr. Rizadi. Just interesting. I was waiting for the yes. Is the explanation, because the mechanism, high activity.
Yes, that's why actually that one is we just run the sample in the SMS and then see what compound is quite obvious in that extract. Actually, we not sure whether that compound is responsible or not. Of course, we need to do the fractionation and then test back to the using the protease inhibition assay again, and after that we do the isolation and so on, and then we will sure which compound is really responsible for that activity. For now, yeah, we just maybe that compound is involved, but we're not sure yet. We just run it under LCMS, even though we're not sure how much of that compound inside. It's very preliminary study. Yeah, of course, after this we will get a grant to study further on that.
Okay, thank you very much. Another maybe like be
Yeah.
in your group.
I'm not sure because I'm not the microbiologist
I see.
actually. But this is the first study that involve actually using the mangrove plants in Sabah. There is study that study the microbe in the soil around the mangrove area. But as far as I know, there's no study about the bacteria in mangrove or whatsoever lah.
So it may be interesting because
Yeah.
maybe we can work,
Yeah, I think it's
collaborate
interesting.
in this area because yeah.
Actually, there is a
Oh, I think another. Any question? Somewhat we will have another lecture with Dr. Vic. Thank you very much again, and I saya kembalikan kepada MC.
Okay, it's very interesting Q&A, right? But we will continue after break. We will have break for 1 hour and see you again at 1 p.m. here, guys. Thank you. Okay, we will have a lunch together here and ada. Okay.
Seksi pengairan.
Cek, cek. Cek, cek, cek. Cek.
Ojo dicokot loh Pak, tapi Pak. Miknya jangan dicokot loh Pak.
Di mana sih?
Check. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. Welcome to our next session. In this session, we will continue our incredible discussion, right? So let me introduce our moderator today, Mr. Ismiarto PhD. He is one of our lecturer in organic field that have research interest in organic synthesis from natural product. So without further ado, let's we are welcoming our moderator, Mr. Ismiarto PhD. Time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Terima kasih atas waktu yang diberikan kepada saya dari MC. Makasih. Dan sebelumnya selamat datang teman-teman dari Sabah, Malaysia, berkenan hadir di tempat kami. Dan juga selamat datang dan selamat siang semuanya untuk ibu dosen ya, satu ibu dosennya, dan bapak dosen, dan juga teman-teman dari program S2 kimia. Ya kan? Pada kesempatan kali ini sebagai tindak lanjutan acara visiting lecturer ya, dan pada sesi siang ini kita kedatangan hadir di depan kita semuanya Bapak Cahyo Budiman, Dr. Cahyo Budiman. Ini perlu diketahui Pak Cahyo Budiman ini senpai saya ini di Osaka University. Yang senpai itu senior saya, walaupun saya lebih tua. Ya, dari ilmunya, dari kelulusannya ya duluan Pak Cahyo Budiman ini asalnya dari Cirebon, seingat saya Pak ya. Ini sok tahu saya, tapi dulu saya pernah ketemu dan ngobrol-ngobrol begitu. Waktu beliau sudah lulus mau kembali ke Indonesia, tapi masih ada aktivitas di Osaka University. Beliau sekarang sebagai senior lecturer di University Malaysia Sabah. Associate Professor. Oke. Terima kasih ya. Dan Profesor Amin ya Pak. Ya Pak, oke. Amin. Oh ya, nggak boleh kampanye ya. Oke. Nggak boleh politik. Udah sedikit nggak apa-apa ya. Dan bisa Bapak Ibu semuanya lihat di biografi ya. Dr. Cahyo Budiman is currently working as a senior lecturer at Biotechnology Research Institute, ya, Universitas Malaysia Sabah. Dan he has obtained his PhD in biotechnology from Osaka University, Japan. His main area of interest focuses on protein science engineering, molecular biotechnology, terus and enzymology. His area of expertise includes protein science, molecular microbiology, enzymology, protein engineering, voltage, molecular biotechnology, chaperone, yeah, molecular biochemistry, and isomerase. And today, ya, beliau sekarang akan membagikan ilmunya pada kita semuanya, itu terkait dengan virtual and fragment-based drug screening for combating malaria. Mudah-mudahan apa yang akan disampaikan oleh Pak Dr. Cahyo Budiman akan memberikan wawasan kita yang lebih, dan mudah-mudahan ada dari kita kerjasama lebih jauh, dan juga mungkin ada mahasiswa dari program studi S2 kita, atau mungkin dari S1 kita yang kemudian bisa tertarik untuk riset bergabung dengan proyeknya Pak Cahyo Budiman. Nah, untuk itu akan dipaparkan secara lebih jelas oleh Bapak Cahyo Budiman. Kami persilakan, Pak.
Baik, terima kasih atas kesempatannya Prof. Meni dan tim Undip atas undangannya ke Semarang untuk kesekian kalinya. Ini saya datang ke sini terakhir sebelum pandemik, Bu ya. Gedungnya sudah baru, jadi agak pangling lihat gedungnya. Dulu kami presentasi pertama di gedung yang lama ya. Ya, gedungnya baru tapi keramah-tamahannya juga masih seperti dulu, masih sangat luar biasa untuk kami. Terima kasih. Tapi memang apa istilahnya ya? Keramahan orang Indonesia itu nomor 1 ya. Mohon maaf yang oleh saya. Baik ya, saya tadi sudah speak dengan moderator, kita pakai bahasa Indonesia saja supaya gampang mencaci maki Malaysia. Mereka nggak ngerti kan? Mumpung di sini kita PSSI selalu menang lawan bola Malaysia kan. Baik Bapak Ibu, berhubung slide saya lumayan banyak ya, jadi untuk hari ini kita coba. Jadi ketika ditanya Bu Mei ini kira-kira topiknya yang, karena kami dari biotek ya, jadi tadi mungkin misalnya kawan saya yang paling muda ya, belah sana baru sunat ya, Dr. Mardani. Itu mencoba mengkaitkan genetik ya, karena background kita kan agak dari biotek ke kimia kan agak lumayan, tapi sebenarnya biotek ini kan area yang luas ya. Dia makanya kita sebut dengan interface sebenarnya, terus kadang-kadang istilahnya juga frontier ya. Ada irisan di situ, biologi, kimia, kemudian molekulernya masuk di situ gitu. Jadi bioteknologi nggak selalu bicara tentang molekular genetik dan sebagainya, memang aspek kimia juga harus kuat. Kayak tadi Dr. Asfa bicara tentang zeta potensial, charging positif negatif, itu kan bukan biologi banget gitu ya. Itu teman-teman di kimia jauh lebih paham. Jadi kita coba bawa riset yang mungkin kita bisa matchkan dengan teman-teman di kimia, karena memang dari sisi background kimia terutama saya memang sangat kurang. Jadi justru disitulah peluang kita untuk kerjasama ke depannya. Ya. Kalau ekstrak gen dan sebagainya kita sambil merem juga bisa gitu ya, tapi ngukur pH mungkin kita masih minta tolong teman-teman kimia. Oke, jadi ini salah satu proyek yang di tempat kami lakukan untuk virtual and fragment based screening untuk salah satu penyakit yang memang banyak prevalence di region kita, termasuk di Indonesia, terutama di Malaysia, untuk malaria disease. Ini pekerjaan yang sudah beberapa tahun juga saya kolaborasikan dengan Prof. Mei. Ya, jadi Indonesia dengan Malaysia dalam status yang berbeda untuk malaria ini. Malaysia hampir dikatakan bebas dari malaria sebenarnya dari tahun 2018. Terutama dari yang disebut dengan indigenous malaria, falciparum, vivax, itu di Malaysia udah kosong. Di Indonesia, di Kalimantan masih banyak untuk falciparum, vivax. Tapi di Malaysia ada tren baru di samping falciparum dengan vivax. Ada spesies parasit kelima, malaria parasit kelima yang emerging dan menginfeksi manusia, yaitu adalah Plasmodium knowlesi. Dan ini juga ada di Indonesia, cuma laporan di Indonesia itu tidak begitu banyak. Saya menduganya karena, nanti saya jelaskan, karena ada masalah teknis untuk diagnosisnya. Ya. Banyak kasus falciparum, kalau kasus P. knowlesi itu terdiagnosis sebagai falciparum. Nah, dan itu banyak sekali. Dan kasus di Malaysia sudah tersetelkan karena ada tekniknya sendiri untuk memberi. Jadi apa itu knowlesi? Dulu kita nggak kenal, dari tahun 60-an nggak ada, sampai kemudian ada orang Inggris menemukan itu, karena awalnya ini menyerang cuma monkey, kelompok makak ya, monyet berekor panjang, ekornya yang panjang ya, bukan yang lainnya. Oke, kalau di tempat kami ngelihat lihat monyet itu biasa aja, masuk ke bangunan, di belakang tempat parkir itu banyak monyet. Kalau di sini kan nggak ada ya, karena kami di dekat hutan kan. Nah itu kenapa kejadiannya? Jadi dulunya dia menyerang monyet saja gitu, tapi kemudian ada kejadian, ada kasus bahwa dia menyerang manusia. Jadi dari monyet kemudian nyebar ke manusia. That's why we call it as zoonotic malaria. Ya. Tentu saja penanganan paling gampang adalah bunuh semua monyet, tapi kan nggak mungkin kan? Ya, jadi tetap kita harus mengobati siapa pasiennya. Jadi ini tren di Malaysia. Untuk yang indigenous, yang biru dia makin turun, tetapi yang merah ini yang zoonotik dia makin naik sekarang 2021. Terakhir saya menguji anak S3 di Sabah juga, dia laporan barunya dia bahkan tahun 2022 makin double kasusnya ya. Jadi ini kasusnya makin tinggi di Malaysia, still istilahnya concerningnya makin mengkhawatirkan, terutama yang terjadi adalah di Malaysia itu di bagian Kalimantan. Dia di Semenanjung juga ada, tapi yang jantungnya itu ada di Kalimantan, terutama di Sarawak, ya di Kalimantan Utara sana, yang dekat Pontianak dengan yang di Sabah. Ya, kenapa sebenarnya? Karena di situ banyak hutan. Dan kasusnya yang paling terjadi adalah di daerah kampung-kampung yang berdekatan dengan hutan, karena mungkin interaksi orang masuk hutan ketemu makaknya ya dan seterusnya itu ini. Meskipun Semarang jauh dari hutan ya. Kali aja jodohnya ketemu di Kalimantan kan, jadi harus hati-hati. Nah ini di Indonesia. Di Indonesia reportnya enggak begitu se-ekstensif di Malaysia ya, tapi Anda bisa lihat banyaknya di daerah yang dekat Semenanjung Malaysia, kemudian di Kalimantan. Kalimantan sayangnya saya nggak percaya laporan ini 2 dengan 3 di Kalimantan itu karena mungkin deteksinya yang menurut saya kurang akurat. Sementara di daerah Sumatera ini wajar karena di daerah peninsular Malaysia juga lumayan banyak. Kita lihat memang jantungnya itu ada di Malaysia. Makanya kenapa kasus Spinolersa ini agak cukup mengkhawatirkan untuk di Malaysia. Jadi karena topiknya SDG ya, apa kaitannya dengan SDG? Ya ini untuk sambal-sambalnya aja ya bumbunya. Jelas berkait dengan poor poverty, public health dan seterusnya. Ini kira-kira jadi pendekatan WHO ya, World Health Organization juga adalah semua kita harus menangani malaria ini dari sisi aspek Sustainable Development Goals-nya dari semua sisinya supaya karena orang sakit nggak bisa kerja, orang sakit nanti kualitas hidupnya menurun dan sebagainya. Oke, jadi ini kira-kira bedanya Knowledge dengan parasite lainnya. Kalau yang lain nyamuk menggigit manusia masuk ke darah ya berkembang jadi zigot, ookinet, ookis dan seterusnya kemudian menyerang hati manusia kan selesai. Kalau ini dia akan menyerang monyetnya dulu, primatenya dulu, seterusnya bersiklus di di dalam darah si primate, kemudian baru ketika dia sudah berkembang nyamuk yang gigit si primate kemudian gigit manusia, itulah terjadi transfer parasitnya ke tubuh manusia. Jadi si parasit itu akan berkembang dulu di dalam primatenya. Baru kemudian itulah kenapa si Spinolersa sejauh ini belum ada laporan dia nyerang manusia langsung. Dia harus membutuhkan host perantara dulu yaitu adalah si primatesnya. Oke, ini agak unik. Nah, sebenarnya obatnya ada. Malaria itu bukan sesuatu yang yang menakutkan banget ya karena obatnya ada. Tetapi isunya adalah resistance. Kayak antibiotik, beberapa bakteri decline efisiensi kemampuan untuk dibunuhnya. Dan itu terjadi bukan cuma di kita ya di Asia Tenggara, tetapi juga di Afrika, tempat-tempat malaria semua. Terutama yang dipakai adalah dari kelompok quinine, meloquinine dan sebagainya. Dia by the time dia akan reduce ya. Jadi treatment untuk Knowledge sekarang ini disebutnya dengan ATC treatment, artemisinin terapi combination. Jadi beberapa obat malaria dikombinasi dikasihkan ke pasien. Sejauh ini dia memang efektif ya. Tetapi karena yang resisten sekarang ini cuma ada di falciparum dengan Vivax. Tapi percayalah, menurut saya ini nggak akan selamanya. Spinolersa pada satu titik juga akan resisten terhadap drug-drug ini. Dan memang sudah terbukti penelitian terakhir yang kali lagi yang hasil studi yang saya uji anak ini yang meneliti ini, dia sudah menemukan polimorfisme pada gen marker yang menjadi penanda resistensi itu pada Spinolersa. Jadi memang parasit Spinolersa evolving, sedang berevolusi untuk menjadi resisten terhadap drug-drug yang diberikan. Itulah kenapa dan kita perlu mengembangkan obat baru yang nggak bisa resisten, yang nggak bisa memicu resistensi di parasit. Nah, apa targetnya? Ini ada satu bahan kimia yang disebut dengan FK506. Saya pikir teman-teman yang di farmasi paham ini. Ini sebenarnya immunosuppressant. Jadi dipakai oleh organ transplantasi. Jadi diminum untuk mensuppress imun sistem kita supaya pada saat organnya masuk tidak direcognize sebagai antigen. Misalnya orang transplantasi liver ataupun ginjal, kalau ginjalnya masuk itu kan semua benda asing masuk akan dikonsider sebagai antigen kan. Nanti imun kita nyerang, ginjal yang barunya itu malah dirusak gitu, di-attack sama imun sistem kita. Nah, jadi orang yang ditransplant itu akan dikasih obat ini supaya organnya tidak diserang, supaya imun kita agak lemah sedikit. Nah, ini bisa membunuh ternyata pinolosai tanpa sejauh ini resistensi yang muncul. Dia terutama dia membunuh di tahapan sizotnya dari si malarianya. Sudah terbukti di falciparum, sudah terbukti di pinolosai. Tetapi kalau kita ngasih obat ini ke pasien malaria nggak akan bisa, karena lama-kelamaan si pasien itu akan makin lemah imun sistemnya. Nanti kayak orang penderita HIV ya, dia punya imun sistem will be out of order. Ya. Jadi bagaimana kita bisa mencarinya? Jadi idenya adalah kita cari pengganti FK506 ini. Dalam drug screening idenya kira-kira kalau molekul itu kan bisa menghambat karena ada satu molekul lain di dalam parasitnya yang dibunuh kan, dihambat, diblok. Jadi kita cari protein yang diblok oleh si FK506-nya, yang kita temukan bahwa namanya adalah FKBP di sini, FKBP 35, yaitu FK506 binding protein, yang sebenarnya secara struktur dia ada dua domain yang berbeda, FKBP, FKBD, di mana FK50 ini akan berikatan, nempel dan kemudian menghambat. Kemudian domain lainnya yaitu TPR, ya, tetra tricopeptide repeat domain, dan CBD ini bukan Cahyo Budiman, tapi calmodulin binding domain. Ya, kita lewat yang itu dulu karena yang paling berperan dalam proses penghambatan ini adalah FKBD-nya. Nah, apa itu FKBP 35? FKBP 35 ini adalah ahli dari yang disebut dengan peptidyl prolyl cis transisomerase, yang di sel ada 3 kelompok, yang yaitu kelompok pertama FKBP, cyclopilin, kemudian parvulin. Mereka kerjanya mirip, sebenarnya adalah untuk mempercepat, mengkatalisis isomerisasi peptida bond selama proses translasi protein, sintesis protein. Misalnya kira-kira begini, pada saat protein itu baru disintesis di ribosom, ya, dia kan prosesnya linear ya, strukturnya itu masih linear, kemudian baru dia folded. Nah, ini teman-teman kimia paham betul. Pada saat dia folded, ikatan peptidanya, ikatan kovalen itu, dia harus ada di 2 kemungkinan konformasi, apakah dia trans ataupun cis. Yang secara energi jelas trans lebih stabil dibandingkan dengan cis. Jadi dia harus muter sampai dia dapat ini trans, selesai. Nah, perputaran ini itu harus secepat mungkin, karena kalau enggak proteinnya akan lambat, slow folding, itu akan mudah terdegradasi. Cuma ada beberapa peptida bond yang dia memang harus dalam kondisi cis isomer, yaitu peptida bond yang sebelum prolin, residu prolin itu biasanya cis, beberapa cis, 20% dari antara mereka cis. Nah, ini karena dia cis, karena energinya restricted ya untuk cis, dia muternya itu jadi lambat. Nah, PPIS itu dia akan mempercepat rotasi peptida bond untuk mencapai cis itu. Kalau nggak ada, dia lambat, dia akan proteinnya misfolding, agregasi. Nah, kasusnya apa? Neurodegenerative disease yang tadi Dr. Madani sampaikan, ada Alzheimer dan lain sebagainya. Itu salah satunya dipicunya oleh karena itu. Jadi PPIS itu memfasilitasi proses ini. Nah, kalau misalnya ini dibunuh, di FKBP 35 dari pinolisanya ini kita blok, maka sintesis protein dari parasitnya nggak akan berjalan, parasitnya akan mati. Karena tidak bisa mensintesis protein secara sempurna. Di dalam sel sebenarnya perlibatannya itu lebih kompleks ya. Kalau kita lihat di sini, FKBP itu dia akan menghambat dia akan mengaktifasi CAN. CAN itu kalsinerin. kalsinerin itu protein yang bisa memfasilitasi, mengaktifasi sistem imun kita. Ya, terutama gen-gen yang berkaitan dengan imunitas, interleukin dan sebagainya. Jadi kalau misalnya kita ngasih obat FK50, FK50 akan berikatan dengan FKBP, kemudian kompleks FKBP dengan FK506 dia akan ngeblok kalsinerin. Kalsinerin nanti dia kalau diblok dia tidak bisa deposforilasi, jadi proses ekspresi dari interleukin gene dan imunnya jadi akan berubah. Ya, jadi pelibatannya itu kalau kita bisa menghambat ini, imun signal dari si parasitnya juga akan terhambat. Nah, kita temukan bahwa di Pinolesi itu juga ada FKBP, namanya yang FKBP 35, lokasinya di kromosom nomor 14, yang dia itu mirip dengan falciparum dengan Vivax. Sorry, ini slide-nya belum dirapikan. Yang juga organisasinya ada dua, yaitu ada FKBD yang merah dengan TPRD. Nah, cuma yang FKBD ini karena yang bertanggung jawab terhadap aktivitas, maka ini jadi daya tarik untuk kita blok. Ya, ini target bagian protein yang harus kita blok. Ya. Vital for regulation PPIS activity. Ya, kalau kita blok nanti semua network activity ini habis, parasitnya akan mati. Kira-kira begitu idenya. Ya, jadi kalsinerinnya juga key regulator. Ya. Nah, cuma gimana kita bisa dapat inhibitor yang lebih baik daripada FK506-nya? Oke. Kita memanfaatkan dua. Yang pertama adalah bioinformatik ya, yang kita manfaatkan tools bioinformatik yang sudah ada. Karena sekarang ini banyak compounds yang berserakan di cloud, ya di database dan sebagainya. Nah, ini yang bisa kita manfaatkan untuk jadi kandidat-kandidat penghambat protein tersebut, kayak malaria box ya dan sebagainya, itu banyak dimanfaatkan. Kemudian yang kedua adalah NMR spektroskopi. Ini makin advance. Sekarang kita bisa manfaatkan untuk screen compounds yang bisa berperan sebagai kandidatnya. Kita masuk yang pertama dulu, mudah-mudahan nggak terlalu panjang. Oke, virtual screening. Virtual itu maksudnya apa? Kalau kita melakukan conventional drug screening, ada 1000 compound di tes satu-satu, jelas akan tadi Dr. Azwa sampaikan sampai kiamat mungkin. Ya, jadi ada pendekatan lain di mana kita memanfaatkan komputer untuk membantu kita men-screening dari 1000 itu di shortlist istilahnya, di senarai pendek. Saya pakai bahasa Melayu. Bahasa Indonesianya apa shortlist itu? Jajahan pintas. Dapat yang beberapa kandidat terbaik untuk kemudian dites secara eksperimen apakah dia ada atau tidak. Dan itu memang sekarang sudah jadi pendekatan umum di industri obat-obatan ya. Ini contoh untuk FKBP 35 yang dari falciparum. Untuk virtual screening dapat kompon ini ya, dan juga dites dengan eksperimental tes, dia dapat penghambatan. Tapi yang melakukan untuk Knowlesi itu nggak ada, karena orang-orang Amerika itu kan nggak aware dengan Knowlesi yang ada di Asia Tenggara ya. Jadi cuma kita yang yang sejauh ini bekerja di Knowlesi. Ya. Nah, apa yang bisa banyak database yang tersedia. Nah, salah satu yang database yang kami manfaatkan itu adalah yang bernama NADI ya, Natural Product Discovery System. Ini database tanaman-tanaman kompon-kompon yang di Malaysia yang ditemukan, yang istilahnya tumbuh di Malaysia. Saya nggak nyebut indigenous atau eksotik ya, yang indigenous ya. Jadi tanaman itu bisa aja tumbuh di negara lain, tapi maksudnya ini semua tanaman yang bisa ditumbuh yang ada di Malaysia. Ini komponnya ada di sini ya, dikembangkan oleh Prof. Habibah, kolaborator kami dari University Science Malaysia. Komponnya sudah ada, strukturnya ada, jadi kita bisa manfaatkan untuk sebagai sumber ininya ya. Dan karena bahan alam ya, kemungkinan untuk aspek safety-nya itu bisa jauh lebih tinggi. Oke, flow-nya seperti apa? Jadi kita preparasi protein yang kita dapat, ya, FKBD sebelumnya kami dapat strukturnya dari NMR, kemudian preparasi dari natural product. Sekarang sampai hari ini dibilang banyak enggak sekarang ini ya, tapi untuk dibilang sedikit juga banyak, karena ini cuma kompon yang ada di Malaysia kan, ada lebih dari 4.000 kompon ya. Kemudian ini nanti dijadikan ligand untuk dock kepada target protein kita. Ya, kita lakukan dock dengan autodock, kemudian dari situ kita screen standarnya adalah FK506 itu. Jadi cut off-nya di situ. Mana yang lebih baik dari FK506 itulah yang kita shortlist-kan. Ya, kemudian dicek stability-nya dengan MD simulation dan sebagainya. Setelah itu baru dites nanti di in vitro-nya seperti apa. Oke, ini hasilnya untuk nadi compounds ya, dari 4.000 kita dapatkan lebih dari 600 ya, punya punya binding affinity ataupun energi yang hampir sama dengan FK506, banyak banget ya. Tapi dari 600 tentu saja kita nggak ambil semua, jadi 600 dari 400 itu cuma 20%-an kan, sisanya lebih jelek dari FK5, kita nggak ambil. Nah dari 600 ini kalau kita shortlistkan lagi, kita ambil 10 besarnya saja. Ini 10 besar yang berdasarkan docking itu lebih bagus daripada FK506 dari punikartin dan sebagainya ya. Cuma kita kan nggak tahu, ini di komputer memang nempel, apakah dia menghambat atau enggak gitu kan. Nah ini harus dites lebih lanjut. Untuk ngetesnya kita harus ngetes untuk dapat kompon ini punikartin kita harus ekstrak dan sebagainya. Cuma sebab saya bukan orang kimia, saya pribadi nggak punya maksudnya secara waktu dan sebagainya kita nggak ngekstraknya. Jadi karena ini proof of concept istilahnya, kita coba dulu apakah memang dari kompon yang tersedia di pasar dari 10 ini apakah bisa menghambat atau enggak. Jadi dari 10 ini kita cek nomor 7, 8 dan 15 ini yang ada di pasar. Nomor 1, 2, 3, 4, 5, 6 ini memang paling bagus, tapi kita nggak bisa tes karena kita harus ngekstrak dulu ya. Jadi kita nggak udahlah nanti aja gitu. Kita coba dulu yang sudah tersedia, yaitu adalah santon, olenic acid. Santon itu dari manggis ya. Olenic acid itu dari Acronites ospera, saya nggak tahu nama lokalnya ya. Tapi itu namanya kayak gitulah ya. Kemudian hidroksiflavon. Jadi dari tiga ini yang kemudian kita tes untuk in vitronya. Nah ini pola interaksinya dari docking ya. Untuk FK506-nya confirm ya, yang merah itu residu-residu yang merah, asam amino yang merah itu adalah residu yang memang sebelum ini kita ketahui itu adalah catalytic sites dia. Jadi kita lihat bahwa FK506 memang ngeblok catalytic site dari si FKBP itu. Memang itulah kenapa memang kompon ini bisa menghambat aktivitasnya. Kemudian yang warna kuning itu asam amino yang kita tahu dia memang memfasilitasi ikatan dengan kalsinerin. Oke, FK5. Jadi itulah kenapa kalau kita tambahkan FK506 dia bisa menghambat juga fungsi kalsinerin, dia bisa menghambat juga katalisis aktivitinya. Oke, interaksinya dominated by hydrophobic interaction. Kalau kita bandingkan dengan santon, ya 3 catalytic site diblok oleh santon, cuma 2 catalytic site yang diblok oleh si yang 2 catalytic site dari 2 residu yang terlibat dalam interaksi dengan kalsinerin diblok oleh santon, tapi dia lebih kuat dari sisi binding energinya. Tapi mirip ya, beberapa yang paling penting untuk kami adalah dia betul-betul menghambat active sites ya. Kemudian juga sama untuk olenic acid ya. Ini bahkan lebih kuat, lebih banyak hydrophobic interaksi terlibat ya. Active site yang diblok juga lebih banyak ya. Dan kalau salah satunya adalah cysteine 105. Cysteine 105, oke. Manusia punya FKBP juga. Similarity dia 70% ke atas. Tapi cysteine 105 ini unik cuma ada di malaria. Artinya apa? Kalau kita ngeblok ini, kalau kita pakai olenic acid karena dia ngeblok cysteine 105, kemungkinan dia spesifik akan ngeblok ke FKBP dari parasite tanpa ngeblok punya manusia cukup besar. Ya, tapi memang harus dites ya spesifitasnya. Kemudian hidroksiflavon juga sama, dia ngeblok beberapa active site yang terlibat ya. Dan kita nggak tahu nih, kadang-kadang kan kalau docking ya kadang-kadang cuma nempel kan. Ya, nempel kan belum tentu, iya kan, orang pacaran juga belum tentu kawin kan. Jadi kita cek dulu stabil nggak nih ya, stabil kan. Jadi kita couple dengan MD simulation-nya ya, kita cek MD molecular dynamic simulation-nya. Ini perbandingan untuk RMSD-nya stabil lah selama 100 nanoseconds ya untuk semua kompon. Girationnya juga oke. Nah, kemudian kalau kita lihat RMSF-nya, residu-residu mana yang quite fluktuatif selama simulasi, hampir residu yang fluktuatif untuk semua komponen ini hampir sama lah, terutama di bagian yang residu 100 sampai 105, yang kalau kita lihat di struktur di sini 105 sampai 120 itu di loop yang warna hijau, ya itu adalah kita sebut dengan active loop, dia tempat substrate itu nempel. Jadi memang dari sisi simulasi kita menduga memang kompon ini memang betul-betul nempel di bagian situ, di area di mana substratnya itu akan dock. Ya. Ini aspartik, ini yang kira-kira RMSF-nya tinggi ya, aspartik 55, 60, isoleucine, tryptophan 77, ini yang terlibat dalam activity ya, untuk catalytic site-nya. Baik, ya, jadi bagian ini adalah yang di loop sebelah situ, dan itu loop yang memang sudah kita ketahui bahwa itu sangat berperan dalam aktivitasnya. Oke, ini struktur detailnya, affinity-nya berapa dan sebagainya dari 4 ini. Santon, affinity-nya strukturnya seperti ini, dan binding site yang terlibat untuk active site-nya yang di blok ada 3, dari aspartik 55, tryptophan 77 sampai phenylalanine 117 dan seterusnya. Jadi kalau kita lihat olenolic acid ini sepertinya mengeblok lebih banyak active site ya dibandingkan dengan yang lain, atau lebih mirip dengan FK506. Tapi kan kita nggak tahu ya apakah memang betul-betul, ini kan komputer ya, bisa saja ini. Oke, kita lihat apakah betul kompon itu bisa mengeblok aktivitas atau tidak. Kemudian kita cek untuk ngeblok, untuk melihat aktivitinya, kita produksi proteinnya secara rekombinan ya. Jadi kita cloning dulu gennya di plasmid, kemudian kita masukkan E. coli, transformasi, ditransform, kemudian pakai heat shock biasa ya, kemudian diekspreskan, normal prosedur untuk molekular biologi. Proteinnya ekspres sangat baik di E. coli, kita pakai E. coli di supernatan. Kemudian kita bisa purifikasi juga ya, dengan ion exchange, nickel anti-chromatografi, kemudian ion exchange, dan terakhir dengan gel filtrasi. Kita dapat yang murni. Nah, inilah yang kita akan pakai untuk in vitro test untuk uji penghambatan. Proteinnya kita dapat banyak sekali, 30 mg untuk 500 ml, jadi cukup, lebih dari cukup untuknya. Kita cek juga oligomerisasinya, dia monomer di confirm di sekitar 16 kilodalton di gel filtrasi. Oke, untuk aktivitas caranya adalah kita menggunakan substrate yang disebut dengan succinyl alanin leucine proline phenylalanine PNA atau ALPF PNA. PNA itu kromogenik ya. Dia kalau kepotong itu akan menghasilkan warna kuning. Jadi isomerisasinya sebenarnya terjadi antara pro dengan leucine. Jadi ikatan peptida antara leucine dengan pro itu akan putar. Nah, kemudian kita tambahkan tripsin. Protease itu bisa motong peptida bond hanya kalau konformasinya di trans. Kalau cis dia nggak akan bisa motong. Jadi idenya adalah dengan assay ini adalah si peptida ini, substrate ini dia akan berputar. Kalau nggak ada FKBP, perputarannya lebih lambat daripada kalau ada FKBP. Bagaimana kita mendeteksi perputaran lambat tidaknya? Kita tambahkan protease. Kalau proteasnya itu berhasil mengcleave bondnya lebih banyak, artinya dia lebih banyak mencapai trans lebih cepat dibandingkan dengan pada posisi yang lambat gitu. Oke, makanya kita sebut dengan protease coupling assay. Jadi assay-nya itu di-couple, dikombinasikan dengan protease. Nah, caranya mendeteksi bagaimana si tripsinnya itu bisa mendegradasi, memotong. Kita detect dengan UV absorbance, karena para PNA ketika lepas dia bisa menghasilkan warna kuning, didetek pada panjang gelombang 390 nanometer. Ini kita dapat ya. Yang biru olenic acid concentration ya, dia catalytic activity-nya drop dengan olenic acid jauh lebih rendah dibandingkan kalau kita compare dengan FK50 warna biru, merah santon. Satu kompon yang tadi meskipun dia hit kompon dalam screening komputer ya, dia nggak begitu menghambat bagus, yaitu adalah 6-hydroxyflavon. Jadi dari screening ini, dari yang tadi komputer ini, dari 3 kompon yang kita ambil, cuma 1 yang behave ya, yang bisa menghambat lebih baik daripada FK50, yaitu adalah olenoic acid. Ini untuk catalytic activity-nya. Kemudian ini IC50-nya kita hitung ya. Jadi 6 hidroksifluorin kita consider nggak ada penghambatan. Xanthan itu lebih tinggi daripada FK50 ya. Olenoic acid dia nggak lebih bagus daripada FK50 tapi agak mendekati daripada 506 ya. Tapi ya memang agak perlu peningkatan untuk ininya, tapi kira-kira kita mendekati ke arah FK506. Kemudian bagaimana penghambatannya terhadap calcinerin phosphate assay ya. Jadi untuk calcinerin idenya adalah sama seperti tadi. Calcinerin itu akan ngeblok dengan fosfopeptid yang awalnya itu dia akan berikatan dengan rodamin. Rodamin itu fluorophor ya, yang bisa menghasilkan sinyal fluorescence. Calcinerin kalau dia aktif dia akan berhasil mendefosforilisasi dari fosfatida ini. Yang kemudian nanti kalau dia defosforilisasi, protease bisa motong, rodaminnya keluar, rodamin yang bebas dia bisa dideteksi oleh fluorescence. Kalau FKBP-nya bisa ngeblok calcinerin, fluorescennya akan rendah karena defosforilisasinya tidak akan berjalan dengan baik, rodamin tetap nempel, sinyal fluorescennya kita suppress. Nah, itu bisa kita deteksi dengan fluorescence di sini ya, pakai eksitasi 485, emisinya 530. Kita lihat di sini konsisten dengan yang tadi ya, hidroksiflavon tidak menghambat, tapi hanya xanthan dengan olenoic acid yang juga menghambat si calcinerin. Jadi confirm ya sejalan dengan itu dan kemampuannya hampir sama dengan FK506. So, jadi kesimpulannya untuk yang virtual ini ya, semua stabil ya, menghambat juga active site dari FKBP, IC50-nya bervariasi, tapi yang paling dekat adalah olenoic acid dengan FK506. Aggregation prevention diabaikan saja. Calcinerin prevention, xanthan dengan olenoic acid bisa menghambat, tapi tidak dengan hidroksiflavon. Jadi ini kami mendapat dari sini kita bisa tahu bahwa memang hasil in silico virtual itu harus selalu dikapelkan dengan in vitronya. Jadi tidak persentase kalau dari kami 1 gagal ya, 1 dibanding 3 itu ya 70%-an Pak ya. Jadi lumayan lah ya, dari virtual itu bisa daripada tadi 4.000 di tes satu-satu kan lumayan juga gitu. Bisa sampai menghabiskan biaya banyak, mending untuk kawin kan. Baik, itu virtual screeningnya. Yang kedua adalah fragment base-nya. Bagaimana sekarang kita nggak pakai komputer tapi pakai eksperimen langsung. Nah, cuma yang kita tertarik adalah pada molekul kecil. Kenapa molekul kecil? Karena senang untuk dimodifikasi. Molekul itu yang disebut dengan fragment based compounds ataupun FBDD kalau nggak salah. Fragment based drug discovery. Ini salah satu bidang yang tengah berkembang. Makanya adalah NMR karena sejauh ini NMR kan kita bayangnya untuk struktur protein, struktur molekul ya, interaksi dan sebagainya. Tapi sekarang kita bisa kembangkan ini untuk high throughput screening kompon yang banyak. Karena cara kerjanya mirip sebenarnya. Karena pada prinsipnya NMR itu kan mendeteksi resonansi dari setiap atomnya kan. Jadi kalau kita bisa kita tahu atom apa yang mau kita detek, ya itu kan kita bisa mendeteksi kalau behaviornya, perilaku dari atom itu sebelum dan setelah interaksi ataupun selama interaksi itu berlanjut. Ya biasanya kalau di NMR yang paling umum kan pakainya karbon, hidrogen, kemudian nitrogen. Ya itu kan semua organik kompon ada ya. Itu semua. Makanya untuk high throughput screening ini agak tricky sedikit. Kenapa? Karena kalau kita targetnya karbon, Anda nggak akan bisa membedakan ini sinyal dari reseptornya atau ini sinyal dari ligannya. Jadi kita harus cari signal yang spesifik, apakah dia spesifik di ligan ataupun spesifik di di reseptornya. Kebetulan di protein itu nggak ada atom F, fluor. Jadi kalau kita mendeteksinya basisnya adalah signal dari fluor signal, dipastikan kalau dapat signal ataupun ada suppressing signal, it doesn't originated from the protein, bukan dari reseptornya. Jadi kita manfaatkan komponnya itu kita tempelkan dengan fluorin ya 31F Nanti kita lihat signalnya itu bagaimana perilakunya without dan with dengan proteinnya. Kalau dia berubah ketika ditambahkan protein, pasti ada something happens di situ kan. Nah, itu prinsipnya. Jadi fluorin ya, ini kalau misalnya nggak ada protein ya, kita dapat signal T0-nya oke. Kemudian nanti kalau misalnya ada ligand binding, dia bisa shifted ataupun bahkan suppress ya. Jadi dengan melihat perilakunya kita bisa lihat ini. Dan simple, kita nggak perlu advance magnetic field dari NMR, nggak perlu 1 tera lawat saya 400 MHz cukup. Ya. Komponnya dari mana? Nah ini yang masih tricky. Nah teman-teman di kimia mungkin bisa mengembangkan lebih jauh. Sekarang di Jepang sudah memproduksi ininya ya. Ini saya dapat dari Osaka soalnya. Kishida itu memproduksi banyak kompon-kompon kecil yang sudah ditempelin fluorinated. Oke, dia udah ada 10.000 lebih. Tapi satu tube-nya, setiap campuran itu satu tube-nya isinya 20. Jadi kalau beli semua library-nya ada 500 tube. Satu tube-nya mungkin kalau di Indonesia kan itu 30 juta. 10.000 ringgit. Ya, jadi kalau mau screening 30.000 ya lumayan. Tapi Bu Meni kaya bisa lah ya. Jual kontrakan 1 bisa lah Bu. Baik ya, kita screen kita cuma pakai 2.000 aja ya karena ketersediaan. Nah ini dari 2.000 ini ya 2.000 maksudnya 2.000 dibagi 20 jadi kami tes 100 tube. Dari 100 tube cuma 1 tube yang ada lumayan hasil ya. Jadi dari 2.000 cuma ini satu peak di sini, satu signal ini adalah satu compound. Bapak Ibu bisa hitung ya, ini ada 20 sebenarnya satu peak yang atas ini. Yang atas itu adalah tanpa FKBP35 ya. Jadi satu peak kita bisa lihat signal fluorin dari setiap kompon. Nah, ketika kita bandingkan sinyalnya dengan ketika ditambahkan FKBP30. Nah, kalau Bapak Ibu lihat ada satu signal yang hilang yang saya tandai di sana dengan PKO 01321. Itu nama komponnya, kode komponnya. Signal itu hilang ketika kita tambahkan FKBP35. Ya, yang lainnya enggak. Jadi dari 1999 sinyalnya masih steady. Tapi hanya satu, ya bayangkan kalau saya stop di 1999 ya, mungkin enggak ketemu. Nah, ini strukturnya ya 2 ring bergabung ya, 3 fluoro and bla 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 ya hidroksifluorid. Nah, yang bawah ini nanti saya ceritakan di belakang. Oke. Sekarang bagaimana kalau saya tambahkan PK0, kan tadinya signalnya hilang ya. Saya tambahkan FK506 ke dalam campuran itu. Ketika saya tambahkan FK506, peak signal dari PKO 01321 muncul kembali. Artinya ketika FK506 itu datang, dia bisa meng-kick out, nendang si kompon yang tadi nempel itu keluar. Artinya apa? Dia nempel di binding site yang sama, tapi binding affinity-nya berbeda. FK50 binding lebih kuat, makanya dia bisa nendang keluar. Tapi untuk kami, it's good things. Kenapa? Yang penting binding site-nya sama. Masalah dia lebih lemah dari FK506, saya minta tolong teman-teman kimia, tolong dimodifikasi supaya nempelnya jauh lebih kuat. Ya, tugas kami selesai di sini. Ya, ya. Fluorinated ya. Fluor NMR betul. Oh. Minus betul. Ya. Oh ya. ppm-nya ini bukan konsentrasi ya. ppm chemical shift-nya. Negatif. Karena nolnya itu standar TMS. Nolnya standar, jadi dia kita bandingkan ke TMS standarnya. Ya. Baik ya. Ya ininya ngelihat signalnya aja ya. Oke, jadi kesimpulannya memang sepertinya dia berkaitan dengan FKBD, tapi afinitasnya lebih rendah dibandingkan dengan FK506 dan berbindingnya di titik yang sama, artinya dia kompetitif ya, kompetitif binding. Apakah betul atau enggak? Kemudian kita lanjutkan dengan titrasi ya. Untuk titrasi itu eksperimennya jadi FKBP 1 tabung ya, ditambahkan dengan ligannya tadi, tapi konsentrasinya beda. kemudian kita 2D mapkan ya antara shifting untuk proton dengan nitrogen. Nanti kita overlay kan signalnya, mana-mana yang shifting, nah itu kira-kira yang residu yang terlibat. Itu. Oh, tadi kenapa TMS-nya itu negatif? Karena kan itu istilahnya T1, T2 ya, dikurangi gitu untuk standarnya itu. Nah, kita lihat beberapa residu memang chemical shift-nya agak lumayan remarkable. Nah, ini kita mapkan yang chemical shift-nya lebih dari 0,2 ppm. Ya. Yang antara 0,1 sampai 0,2 itu yang warnanya magenta, kemudian yang 0,2 yang lebih besar itu yang merah. Ini semua residu ini ya, E139, isolation 95, sustain 126, ini semuanya adalah residu-residu yang memang di catalytic site. Jadi memang confirm si kompon itu nempel di bagian catalytic site yang bisa kemudian menghambat aktivitasnya. Kita bisa kalkulasi juga untuk KD value-nya ya. Jauh 400-an, ratusan ini ya, angkanya mikromolar. Kalau kita bandingkan dengan FK5 memang 10 kali lebih rendah. Ya, jadi kenapa sebenarnya lebih rendah? Karena molekulnya kecil. Kalau Anda lihat strukturnya itu cuma 2 ring struktur ya. Sementara FK506 itu macrolide itu besar banget. Ya, jadi wajar ininya apa namanya? Aktivitas lebih penempel afinitasnya itu lebih rendah, dan kalau kita bandingkan penghambatannya FK506 menghambat lebih bagus daripada PK 01321. Tapi kita at least dapat satu ini bagus karena memang kemudian kita tugas berikutnya adalah memodifikasinya, tambah fungsional group atau growingnya ya, atau di link dengan yang kompon yang lain dan sebagainya. Kita sudah coba juga untuk mendapatkan kompleks strukturnya dengan FKBD, kita try untuk kokristalisasi tapi sampai sekarang yang belum berhasil. Kristalnya dapat tapi kualitasnya jelek, dan kemudian grant kami selesai, jadi nggak bisa lagi jalan-jalan ke Jepang. Mudah-mudahan dengan Diponegoro bisa kita lanjutkan. Oke, jadi kira-kira seperti ini ya. Jadi kita bisa memanfaatkan virtual screening, ya. Semua kompon sepertinya bisa berinteraksi dengan melalui hidrofobik. Yang paling bagus adalah olenic acid. Kemudian untuk fragment base kita dapatkan PKO 01321, tetapi masih lebih rendah dari FK506. Next works is how to improve acid. Tapi ini juga salah satu keberhasilan bahwa fragment fluorinated screening dengan menggunakan NMR bisa kita pakai untuk malaria, bahkan juga untuk penyakit-penyakit yang lain. Dan saya pikir bisa dilakukan di Indonesia karena kita nggak perlu yang canggih banget NMR-nya. Yang penting kita 400, kalau bisa ada cryoprobe. Itu aja sih. Ya. Oke, saya pikir itu ini supporting grant kami dari universitas kami, juga dari Osaka University. Ini tim yang terlibat, Bu Meni juga dalam penelitian malaria ini. Ya. Student saya, Iliana ini yang sekarang lanjutin project, Jovi sudah selesai, Prof. Li Pingchin, dari Osaka ada dari Tambak Ukeng, dari Protein Research Institute ada Prof. Fujiwara, kemudian dari UPM. Kemudian itu saja. Ini mahasiswa grup saya, 2 mahasiswa saya dulu ke Undip nih, diajarin juga apa namanya Pak Azari ya, ada fotonya tuh Pak. Ya, dulu berdua di sini. Terima kasih Undip juga numpang di asramanya yang kata mahasiswa saya kayak hotel. Kemudian ini grup kami yang masih eksis sekarang ya, yang Iliana itu yang pakai jilbab biru di belakang yang masih kerja dengan FKBP 35. Saya pikir itu saja. Terima kasih semua. Assalamualaikum warahmatullahi wabarakatuh.
Menarik sekali paparan dari Pak Cahyo terkait dengan apa ya, eksplorasi ya Pak ya, eksplorasi sebagai pengganti FK506 gitu ya. Nah, yang kemudian eksplorasinya bisa melalui dua cara ya Pak ya. Yang pertama dengan dengan screening ya, mungkin dari teman-teman dari mahasiswa yang juga mengikuti tadi, ada yang menarik kemudian untuk dikembangkan atau nanti ikut join project Pak Cahyo, itu bisa memberikan pertanyaan atau komentar ya supaya kita lebih paham lagi mengenai apa yang disebut Pak Cahyo. Kami persilakan mungkin dari Bapak Ibu dosen atau dari Mas. Kami persilakan. Ya, tanyakan suhunya lah. Oke, silakan Pak. Ya. Terengganu, Terengganu. Langsung saja dijawab. Kayaknya beberapa item.
Ya, yang pertama ini itu apa? Kalau Yasara masalahnya harus banyak.
Dari teman yang lainnya, ada yang komentar? Silakan.
Terima kasih atas waktunya. izin bertanya kepada Bapak Dr. Cahyo. Jadi terkait proses MD juga, biasanya kita kan setelah melakukan MD akan melihat hasil interpretasinya itu. Nah, biasanya kan ada kita melihat energi interaksi yang paling rendah dan biasanya itu yang akan kita lanjutkan untuk docking ke proses selanjutnya yang akan jadi ligand ataupun reseptor dan yang akan kita compare menjadi kompleksnya kita gitu. Nah, biasanya kita memilihnya itu berdasarkan dari grafik itu kan yang paling rendah gitu. Jadi berdasarkan yang sudah Bapak jelaskan sebelumnya, kemungkinan meski dia energinya yang paling rendah, tapi bukan dia yang akan belum tentu dia yang akan menghambat seperti itu. Nah, kira-kira kalau misal kondisinya waktu kita in silico, kita memperoleh untuk mencegah proses in vivo secara wet lab-nya itu sesuai, kira-kira cara pemilihannya bagaimana? Biasanya kan itu banyak hasilnya gitu, Pak. Oke. Iya. Ya. Terima kasih Pak atas jawabannya.
Oke, untuk yang lainnya mungkin masih ada satu kesempatan teman-teman untuk memberikan pertanyaan. Yang deretan belakang, mungkin ada yang perlu ditanyakan. Iya, oke, silakan. Perkenalkan dulu, mungkin namanya biar.
Halo.
Online juga.
Apa-apa. Oke, tidak keluar suaranya. Baik. Baik, untuk yang di binding affinity, ini kan yang dilihat itu Gibbs energinya kan, energi bebasnya. Jadi kenapa kita cari yang paling negatif, karena secara konsep kan ya, kalau molekul lebih stabil kan energi bebasnya kan lebih paling rendah kan ya kan, itu kan energi yang tersedia untuk bekerja kan, energi bebas itu. Kalau banyak energi bebasnya nggak stabil, kita bisa ngegosip, bisa ngapain gitu, nggak stabil. Ya jadi apa kalau kompleksnya itu benar-benar eh, natural, itu kan harusnya energetically favorable ya istilahnya ya. Secara energi dia itu membuat dua molekul itu nempel secara nyaman. Kalau dia nyaman berarti energi bebasnya itu harusnya sih paling rendah gitu. Jadi eh, sebenarnya saya nggak begitu yakin ada standar berapa paling minimum. Yang paling jelas sih cut off-nya adalah apa ligan yang kita jadikan sebagai standarnya itu. Yang penting dia lebih rendah dari situ menurut saya sih nggak ada masalah gitu. Apakah secara eksperimen binding affinity itu merefleksikan KD ataupun kayak menurut saya sih enggak. Dan apakah mungkin teman-teman kayaknya lebih paham kalau di enzim kan konstanta Michaelis-Menten KM kan makin rendah KM sebenarnya makin bagus kan karena catalytic efficiency kat per KM-nya akan jadi lebih tinggi kalau KM-nya rendah. KM rendah itu kan artinya bisa saja dia kan berapa jumlah substrat yang diperlukan untuk menginisiasi reaksi kan, atau juga itu kan kadang-kadang orang merefleksikan juga itu binding affinity kan. Kalau binding affinity-nya kayak seolah-olah lemah, kiket per KM-nya jadi bagus. Kalau KM-nya tinggi, berarti kan dia nempelnya kuat banget ya. Itu kan kiket per KM-nya jadi ngedrop gitu loh. Jadi kadang-kadang strong binding affinity belum tentu dia bagus untuk catalytic activity. Kenapa? Karena kalau substratnya itu nempel terlalu kuat juga nggak bagus kan, substrat harus dilepas kembali kan kalau di enzim. Cuma itu kalau kita bicara substrat, tapi kalau inhibitor kita pengennya yang nempel terus kan. Jadi itu memang hubungannya agak sedikit lebih susah ya. Apalagi karena parameter perhitungan di komputasi untuk binding energy dengan parameter perhitungan di tingkat eksperimen itu berbeda. Jadi kadang-kadang memang nggak lari, nggak sejalan gitu. Ya. Mudah-mudahan menjawab Pak. Ya. Pak Azhari mungkin bisa nambahkan. Ya.
Atau mungkin ini Pak Cahyo, jadi seperti di kuliahnya Pak Asari, nanti ada 1 atau 2 sesi pertemuan, nanti bisa diisi oleh Pak Cahyo jika memungkinkan. Jika nanti kan ada semacam guest lecture, tapi bisa jadi online, itu juga bisa lewat dengan Microsoft Teams. Jadi ada banyak diskusi yang mungkin bisa jadi sangat menarik, terutama bagi teman-teman sampai ke aplikasinya. Itu. Oke.
Tapi kan katakanlah misalnya komorpul saya tadi berhasil di
Ya, terima kasih banyak kepada Dr. Cahyono dan Pak Ismiarto yang telah memandu sesi Cahyo. Dr. Cahyo Budiman dan Bapak Ismiarto. Oke. Mungkin kita sampai di penghujung acara untuk studium general hari ini. Terakhir, saya sebagai MC mohon maaf atas segala kekurangan. Untuk acara selanjutnya akan dilanjutkan untuk from group discussion setelah sesi ini. Untuk sesi studium general, mohon maaf atas segala kesalahan. Saya akhiri. Assalamualaikum warahmatullahi wabarakatuh.